Welcome everyone to the 16th annual Judd Symposium plenary session. This Judd Symposium is our departmental showcase of our early career investigators, originally launched by our former chair from 1977 to 2014, Dr. Louis Judd. During his remarkable tenure, Dr. Judd oversaw the development of the UC San Diego Department of Psychiatry as one of the premier departments in the world. For those that know Dr. Judd, he was passionate about education and aimed to create an environment where clinicians, educators, preclinical and clinical scientists, trainees, and faculty can interact to enhance cross-fertilization within the department. To recognize our trainees in support of this goal, he inaugurated this early career research symposium in 2005 to provide a forum for our trainees to showcase their work and to receive feedback. In 2014, this symposium was named in his honor. In a moment, I'll pass along the Zoom controls to Dr. Pat Judd to tell us more about Lou. Then we'll go to our chair, Dr. Jeff Daskalakis, to kick off the event. Then finally, we'll hand it over to our Master of Ceremonies for the rest of the event, Dr. Yash Joshi, last year's keynote speaker. This year, we had nearly 100 submissions for the Judd Symposium, including many who indicated a willingness to be considered for a talk in this plenary session. These submissions were reviewed by a program committee comprised of faculty and members of the Trainee Council, a process that was once again led by Dr. Neil Swerdlow. As many of you know, for the past 7 years, Dr. Swerdlow has served as our Department Vice Chair of Education and has been a leader in developing our educational programs, but is now stepping down from this role. While we're here, we'd like to take a moment to thank Neil for his efforts with all things EDU, including his tireless efforts with this year's Judd Symposium. We'd also like to take a moment to thank Jennifer Hickey Hinojosa for her outstanding leadership with PET, and Sean Rybeck for his wizardry with the Zooms and the IT aspects of the department. Today's plenary session features the 8 highest rated abstracts, including this year's keynote speaker, Dr. Monica Fedorov. Before I pass over the Zoom mic to Professor Emeritus Pat Judd, I have one last announcement. Unfortunately, we cannot follow our tradition of an in-person poster session that immediately follows today's talks due to COVID once again. The poster session was previously a great opportunity for our department to connect around the science of our early career investigators. Following the recommendations of the Trainee Council, part 2 of this Judd Symposium will be Tuesday, March 16th via Zoom from 9 to 11, and will feature several concurrent mini symposia. We've asked the participants to still imagine this upcoming event as an in-person poster session with 5 minutes describing the research and another 5 minutes fielding questions and discussions about the work. Since we now have limited options to break out of our Zoom bubbles, please make an extra effort to carry on the tradition of connecting with one another and save the time slot for March 16th for part 2. Let me now pass over the Zoom to Dr. Pat Judd to tell us about the person who built and led this great department for the better part of 4 decades, Louis Judd.
Am I on? Okay, hi. I'm looking at Lou as I speak, okay. So anyway, thanks, Greg. And just a minute about Greg. I met Greg in the 90s. He was a JDP student, and he did his did his clinical internship at the Gifford Clinic, and then he went on to become a psychology intern in the department. Of course, now one of our preeminent faculty members. He's an example of a wonderful research career within the department. Lou would be so happy and proud to be here today and see how the symposium has grown and developed. It speaks to a central part of his legacy, which was to generate research into the causes of and contributors to serious mental illness. Lou became a psychiatrist when the educational emphasis was on clinical care. one-on-one clinical care. And he was devoted to that care and was still seeing patients prior to his retirement. He trained at a pivotal moment in the history of treatment of the mentally ill. Patients who had been warehoused in large state hospitals were being released into the community. They had freedom, but minimal treatment. And we actually still see the results of this today in our large homeless population. Treatment with medications was just beginning. Prior to that, there'd been insulin shock, ice baths, psychotherapy, psychoanalysis, which did not prove effective with the seriously mentally ill. He knew that the answers lie in research. His first paper was a case study of a child with obsessive compulsive disorder. Lou trained in both adult and child psychiatry and had his own psychoanalysis. You know, in the 60s, psychoanalysis was still a core feature of all the training programs in psychiatry departments. He met Arnold Mandel, the first chair of the department, in the 60s, and there was immediate synergy between the two of them. Arnold wanted to build a department based on the neurosciences. And Lou, so he had that vision, and Lou realized that vision through developing a strong research department and also through launching the Decade of the Brain when he was director of the National Institute of Mental Health. One of Lou's great strengths was his ability to recruit the best psychiatrists and psychologists in the country. His longest recruitment took 6 years. He was a relentless recruiter. He wined and dined people, stayed on top of it. It was really endless. And actually that person is here today and is one of our premier psychiatric faculty. However, Lou was also a Renaissance man. He was a gourmet cook. He loved the arts. He played tennis. He ran daily. And he was a rabid football fan and a world traveler. We had a rich family life with our girls and then our grandchildren, and he enjoyed his life. And that should also be a legacy to you young researchers to lead a balanced life and enjoy yourself along with your work. Okay, now I look forward to hearing your presentations. Thank you.
Thank you, Pat. It's my tremendous honor to serve as chair of the department, of this great department, which is known for its exemplary research that starts with the early career investigators, many of whom continue on to academic careers and many of whom are presenting today. UCSD Departments of Psychiatry Research Future is served by bright, highly motivated individuals who are guided by world-class mentors that are equally motivated and supportive by mentors throughout their career and who shine the spotlight on the trainees as is being done today. I'm excited to be part of this rich tradition that Lou Judd started, which is now being supported by previous leaders, including not only Drs. Grant and Antonelli, but also Neil Swerdlow, Greg Light, the PET Council, PET Trainee and Leadership Council, and of course, our Research Council. I want to tell you that my commitment as chair is to be a champion to the development of future leaders in mental health. This is very important to me, and symposia like these are proof of the fact that UCSD Psychiatry is an incubator of exceptional world-class talent. I've reviewed the submissions and I'm thrilled and impressed by the caliber of work which stands toe-to-toe with any other great department of psychiatry. I'm very much looking forward not only to today's talks, but also the mini symposium in March that Greg mentioned. I request that everyone do their best to attend. I'm now happy to kick off the start of today's session and hand it over to our MC for today's events and last year's Judd keynote speaker and now assistant professor in our department, Dr. Yash Joshi. Yash, over to you.
Thank you, Dr. Daskalakis. Now we get to what we've all been waiting for. You can see, if Sean advances the slide, the agenda for the day. We'll have 4 15-minute talks followed by a short break, followed by 3 15-minute talks and a final 20-minute plenary. After each talk, there'll be up to 5 minutes for follow-up questions. For everyone in the audience, please keep your microphones muted throughout the speaker's talk. We'll be using the chat function on Zoom to coordinate questions for our speakers. You can either post the question in the chat and I'll read it out loud, or you can say the question, say that you have a question, I'll identify you and you can unmute yourself and you can ask the speaker directly. We will try our best to stick to the schedule, but it is entirely possible that we may not have time for everyone's questions. We encourage you to reach out directly to the speakers for follow-up. After all, connecting with others is one of the big draws of the Judd Symposium. And so without further delay, I would like to introduce our first lead-off speaker. Dr. Aaron Besterman is an assistant professor at UCSD and serves as an attending psychiatrist on the Child and Adolescent Psychiatry Service at Rades. He's also a clinical investigator associated with the Rades Children's Institute for Genomic Medicine. Dr. Besterman completed residency at UCSF, followed by child fellowship at UCLA prior to coming to San Diego. He has previously received the ACAP Junior Investigator Award among numerous other distinctions. Today, Dr. Besterman will be presenting his work titled Functional Instructional Analyses of Novel Smith-Kingsmore Syndrome-Associated Mtor Variants Reveal Potential New Mechanisms and Predictors of Pathogenicity. Please welcome Dr. Besterman.
Thank you very much. Let me go ahead and share my screen. Hi, everyone. My name is Aaron Besterman. Thank you very much for that kind introduction, Dr. Joshi. So I'm a new faculty member in the Division of Child Psychiatry here. And I'm really honored to be able to present some of the research from my postdoctoral fellowship at UCLA to everyone here in the department today. So I don't have any conflicts of interest to report. So I study neurodevelopmental disorders, and just to make sure that everybody is on the same page about what neurodevelopmental disorders are, I'll just go ahead and define them for the purposes of this talk. When people talk about neurodevelopmental disorders, they typically are referring to autism spectrum disorders, which are disorders characterized by deficits in social communication. So people on the autism spectrum typically have difficulty understanding social cues and understanding how to socialize appropriately. And they also have what are known as restrictive and repetitive behaviors. So these might be things like hand flapping, lining up toys, or having a very rigid mindset about how things should be done. Global developmental delay is another neurodevelopmental disorder, and very generally, this refers to not reaching developmental milestones on time. And lastly, for the purposes of this talk, intellectual disability is another neurodevelopmental disorder, which is generally defined by deficits in intellectual and adaptive functioning. And so when I refer to neurodevelopmental disorders, these are the things that I'm going to be talking about. And what we've learned over the past decade or so by the work of many dozens of people around the world and with a lot of effort is that there's a large genetic basis to neurodevelopmental disorders. And at this point, many dozens of genes have been identified to be associated with these neurodevelopmental disorders. And these genes tend to cluster into different biological functions, as you can see, to gene expression regulation, neuronal communication, and so forth. And if we take a close look at what these genes do, what we notice is that they tend to fall into similar pathways, signaling pathways. And one of those signaling pathways that some of these genes cluster into is known as the PI3 kinase AKT mTOR signaling pathway. And this is a really crucial pathway in many cellular processes, both in neural development and also in the growth of cells throughout the body. What this pathway is important for is detecting external factors that send signals about the nutrition and environment outside of the cell, and then controlling cellular growth and metabolism. And what we've learned is that disruption to this pathway, and primarily activation, changes in the genes in this pathway that cause it to be overactive, can result in a lot of neurodevelopmental overgrowth disorders. So when I arrived at UCLA, I started my postdoc with Dr. Julian Martinez, who's a medical geneticist, and he was studying the diseases caused by mutations in this pathway. And so an example is mutations to the PIK3CA gene and this causes related overgrowth disorders. And as you can see in this child, the mutations are just in the child's leg and foot, and they're not in the cells throughout the rest of the body, and so you can see excessive growth there in a syndrome caused by mutations in the PTEN gene. It results in PTEN hamartoma tumor syndrome, and this happens throughout the body, and you can see excessive growth of this child's head. And then also, as you might expect, activation of this pathway can lead to increased rates of cancer. And so people with this disorder have elevated risks of cancer at different organs and need to be monitored for that. Mutations to AKT cause a syndrome known as Proteus syndrome that can really be very disabling. And many of you may have heard of tuberous sclerosis complex caused by mutations to TSC1 and TSC2, and these are cortical tubers or growths within the brain. And so when I arrived, there were patients in the clinic who had all of these different disorders. And just recently, there were a couple patients who had been recruited where there were actual pathogenic variants or mutations in this Mtor gene, which is sort of the downstream receptor of this whole signaling pathway that then regulates a lot of the cellular growth. And so this was sort of a newly described disorder, and I was really interested in that. So if we take a close look at the protein of Mtor protein here, these are two protein domains, or three, the fat domain, kinase, and fat C domain. And what you'll notice is that these are different mutations within these domains that result in different Mtor related disorders. So depending on where the mutations are in the body, how they're distributed, whether it's mosaic only in a small fraction of cells or throughout the body, you get different disorders. So if it's just in a tiny group of neurons in the brain, you'll end up with something known as focal cortical dysplasia type 2, which basically means that the normal, very nice layering of the cortex doesn't occur as it should, and you get this really disorganized, chaotic cortical structure that usually results in seizures from a small change in the brain. And if you look at the blue mutation, this results in a syndrome known as asymmetric megaloencephaly with linear hypommelanosis. What that basically means is in the parts of the brain that have the mutation, it grows too much. In this case, just half of the brain. And then in the areas of the skin where the mutation occurs, you have hypopigmentation. But if the mutation is present everywhere, you get this syndrome known as Smith-Kingsmore syndrome. And this is a photo of some of the kids that we've recruited. And you can notice that there's overgrowth, that their heads are larger than normal. And this had just been described when I started my postdoc. And this was one of the early sort of descriptions of the disease. And what you'll notice is that there's sort of two key neurobehavioral traits that I was interested in. These kids almost universally have intellectual disability. They also have large heads and oftentimes seizures, and about a third of them have autism. So I was really interested in studying these kids in more depth. So we started recruiting kids over the past couple of years. We recruited about 17 kids around the world to participate in the study, and their families completed questionnaires. And 7 of these kids with Smith-Kingsmore syndrome had novel variants that had never been described before. So these are our 7 patients. These are the novel variants. You'll notice they have similar features to what I described before: large heads, developmental delay, intellectual disability. And then we describe some new features which had not been described before: constipation, recurrent infections, asthma, abnormal corpus callosal development, and anemia. And there was one patient, the oldest one that I'm aware of, at 52 years of age, who also had intraosseous meningiomas, which is a very rare brain tumor where it invades the skull, and then also cognitive decline and potential dementia, which had not been described before. Given it's only one patient, we don't know if this is part of the disease, but it's concerning and something that we want to continue to monitor for in subsequent patients. What we wanted to know was, how did these mutations actually result in disease? So there were a lot of potential mechanisms, by which the mutation could cause pathway activation, but we wanted to explore this and confirm the sort of theoretical mechanism. So we collaborated with Dr. Mark Nellis, who's a molecular biologist in Erasmus University, and we did some molecular studies to look at downstream signaling pathways. And so we looked at this mTORC1 complex and the phosphorylation of its downstream target, S6 kinase, compared to control. And the red arrows indicate the novel variants, and the other bars are the sort of other variants we were interested in. And what you see is that there's increased phosphorylation compared to control in the novel variants and some of the other associated variants we were studying, suggesting that activation of this mTORC1 protein complex is one mechanism by which the mTOR mutations may cause overgrowth. And then, but there's an additional part of this signaling pathway known as mTORC2. mTOR can partner with other proteins and cause activation through this complex as well, in theory. So we studied a target of this complex known as AKT, and when it's phosphorylated at this site, it causes activation. So we looked at the amount of phosphorylation at the serine 473, and what we see is a small amount of activation with certain mutations, not as dramatic as mTORC1, suggesting that potentially mTORC2 signaling is a secondary, smaller mechanism by which activation occurs. And this had not been shown before. mTORC1 had been demonstrated previously. And so this might seem very minor, but it actually has a lot of clinical implications. So it's been well known for a while now that if you inhibit mTORC1, and this has primarily been done in cancer, if you inhibit mTORC1 with what are known as rapalogs, these are medications like rapamycin, and they uniquely inhibit mTORC1, what happens is there's these positive feedback loop mechanisms that cause activation through mTORC2 and ultimately can result in additional cell proliferation and survival. And so there's a lot of patients now who are sort of seeking out treatment with rapamycin because of the theoretical possibility it could inhibit mTORC1 and stop cellular growth. But we have a lot of concerns that it could potentially activate this positive feedback loop through mTORC2. So one might want to look for inhibitors that target both of these pathways. So an interesting observation, now that we sort of have some knowledge about the signaling mechanism, is that if you look at this protein carefully and look in databases where you can find genetic variation in unaffected individuals who don't have disease, you see a lot of genetic changes all around here that don't cause disease. So what we wanted to try to understand was, why did these specific genetic changes cause these overgrowth syndromes and neurodevelopmental disorders, and nearby changes not cause any disease? And so we worked again with some collaborators, Jeff Abramson and his postdoc Thornton Hoff at UCLA Department of Physiology, who are structural biologists, and we took a closer look at the 3D structure of the mTOR protein to try to understand this a little bit better. And what this is mTOR, and mTOR rotated 90 degrees, so you can see both sides. The green here represents normal variants that we see in the general population. Purple is variants associated with overgrowth, and the orange are variants associated with cancer. And what you see is that the cancer and overgrowth variants tend to cluster in the middle, near the catalytic site of the protein. And if we zoom in and take a closer look, what you can see is that the purple variants associated with overgrowth really tend to point inward towards other residues. These are known as alpha helices, and they pack together in the center of the protein. And what you see is, even though there are nearby benign population variants, they tend to face externally, not inside towards the other alpha helices. So we thought maybe that these pathogenic variants tend to disrupt these protein residues that point inward inside the protein. But we wanted to try to confirm that by looking throughout the protein in sort of a quantitative way. And so we found this measure known as relative solvent accessible surface area. And what this basically means, it's basically a measure of the surface area of a biomolecule that is accessible to solvent. So if the red is the physical surface of a molecule, the accessible surface is sort of the dotted line around it. How much can solvent get inside? And if the variant is on the deep inside of the 3D structure of a protein, the solvent's not going to get in there very well, and there'll be a low, what we call RSA for this term. And if it's on the outside, there'll be a high RSA. And so we wanted to look throughout the protein and compare the RSA levels, which is on the y-axis, between these different groups, the green being population, orange known cancer variants, the purple known neurodevelopmental overgrowth variants. And then these are the subtypes of overgrowth disorders. And what you see is that there's a significant difference between population variants. They're more external, having higher RSA, and then cancer has a lower RSA value. And lastly, the neurodevelopmental disorders, those are the most internal variants, and they have the lowest RSA. And so when we then said, okay, so how good is RSA at distinguishing between pathogenic and non-pathogenic variants? We saw that it's sort of moderately accurate in predicting the difference. This is known as a ROC curve, and we basically compare the sensitivity to the specificity of this measure. And what we get these area under the curve values, which are basically a measure of accuracy. And what we see is that RSA is moderately accurate at distinguishing between neurodevelopmental disorder variants and cancer variants from population variants. And so in conclusion, what I've described is that the phenotype of Smith-Kingsmore syndrome is broader than previously known. SKS-associated variants may activate overgrowth via mTORC2 in addition to mTORC1, which has important treatment implications. Pathogenic mTOR variants tend to lie deep in the core of the protein, disrupting alpha helix packing. Relative accessible surface area, a quantitative measure of solvent accessibility, is lower for the pathogenic mTOR variants than for the population variants. And relative accessible surface area moderately differentiates pathogenic mTOR variants from population variants. And my last slide, some future directions. We'll be studying how RAS compares to other predictors of pathogenicity in mTOR, trying to understand if RAS measures are correlated with specific phenotypes in SKS, and is this RAS measure predictive of pathogenicity in related overgrowth disorders, not just Smith-Kingsmore syndrome. And so thank you very much for your time. I really want to thank the Smith-Kingsmore syndrome patients and families who participated in the study. A very large thanks to Dr. Julia Martinez, who's been my postdoctoral mentor throughout all this, and then my collaborators and everyone here for listening to my talk. Thanks very much.
Thank you, Dr. Besterman. We have time for one question.
I have a question, Yash. This is Susan Powell.
Go ahead.
I was just wondering, are you considering making a mouse of this disorder? Or has anyone?
So I am not considering making a mouse. I do know of one group in Japan who has made a mouse pretty recently of this disorder.
Do they show the overgrowth phenotype?
They haven't, as far as I'm aware, they haven't described that yet. It was more of looking at sort of neurophysiologic effects.
Uh-huh. That'd be interesting. Thank you. Great talk.
All righty. Thank you, Dr. Besterman. While we get the next speaker ready, I will introduce her. We have Dr. Lieselot Caretti, who is currently an assistant project scientist in the Department of Psychiatry. working with Dr. Olivier George. Dr. Coretti received her PhD and master's degree from Ghent University. She's been a researcher and scholar at multiple institutions, including the Scripps Institute, Massachusetts General Hospital, and Harvard Medical School. She'll be presenting her work on the interplay between cocaine and the feeding hormone leptin in genetically diverse rats. Please welcome Dr. Coretti.
Good morning. Thank you so much, Yash, for the kind introduction, and thanks to the organizing committee for giving me this opportunity to present. I'm going to start this presentation off a bit backwards with the acknowledgement slide, just because it shows you what I am in the department. I'm in the George Lab, and so I want to acknowledge Olivier George as my mentor, and then some additional people who were important in this project, Professor Guglielmo, Molly and Brent for technical support, and my interns, Christina and Caitlin. Overall, the George Lab works on preclinical addiction research, and we contribute this in a collaborative project with Professor Palmer and Professor Solberg-Woods for genome-wide association studies of addiction-like behavior in rats. And this project looks a bit as follows. So the Solberg-Woods lab contributes heterogeneous stock or HS rats. This is an outbred rat population that is generated from 8 inbred rat strains that over many generations through careful breeding are generated and result in this very diverse, genetically diverse population, which actually mimics the diversity found in humans. And so with genetic diversity comes phenotypic diversity. And so our contribution in this project is to characterize the addiction-like behavior of these animals through models of self-administration in rats. And then this goes through to the Palmer lab, who does whole genome sequencing of these animals and does the analysis to then find phenotypic loci that contribute to vulnerability or resilience to addiction-like behaviors. So how we do this behavioral characterization, we have a standardized protocol, which starts with a surgery that will insert a catheter into the jugular vein of these rats that allows for IV drug administration. And after recovery, the rats will be put in these self-administration boxes where they have access to a lever that they can press to give themselves these drugs through IV. And so they do that initially in 2-hour sessions a day for about 10 days, and we call that short access. And then after some time, we extend their sessions to 6 hours, we call this long access, and they do that for 14 days. And so a typical development of addiction-like behavior is that they will escalate their intake. So you can see that over the sessions, they will press the lever more to give themselves more drug. Another addiction-like behavior that we test is motivation. And we can do that through these PR tests, which stands for progressive ratio. So you put the animal in the boxes, and instead of 1 lever press for an infusion of cocaine, they have to press a progressive amount of times for the next infusion. And so it causes more effort, and this allows us to test motivation. The last typical addiction-like behavior that we test is compulsivity, and we can do that by pairing an infusion of drug with a electric foot shock that we deliver through the floor grid. And so the fact that these rats will continue pressing despite this adverse consequence measures their compulsivity. And now to really characterize these animals, we introduce a new variant, which is the addiction index, and can be calculated through the average of disease scores of these different measures. And so it's a new variable that is available for every animal, and which allows us to group the animals in a group with high addiction-like behavior if they have a high addiction index, or a low addiction-like behavior group if they have a low index. And if we do that for these different measures, we can see that in this population, we actually have 2 groups of animals, one that is that is vulnerable to developing addiction-like behavior, and one that's more resilient. And they are actually significantly different in all these behaviors. After the rats are completely characterized, we will divide them into 3 groups for sacrificing with drug on board, in withdrawal, or while they are in abstinence. And the reason we do that is because we actually keep these tissues. They're very valuable because they are genetically and behaviorally well characterized. And we combine these tissues with other samples that we take throughout this timeline of urine, blood, and fecal matter. And we store them in different ways in this biobank that we make available to other researchers who want to do follow-up studies. And these follow-up studies are actually very useful because HS rats allow us to link this phenotype of high or low addiction-like behavior to the underlying molecular and cellular differences. And about a year ago, we put out this paper in which we saw and showed that rats with a high addiction-like behavior actually have a higher GABA transmission in the central amygdala, which is related to a lower level of nociceptin, which is an endogenous opioid-like peptide. So we can actually characterize these rats. Now, the rest of this presentation will be based on an observation of body weight changes in abstinence. And what we see is that males that have a high intake of cocaine actually have a lower body weight significantly compared to low intake rats and naive rats. On the other hand, females don't have this weight loss. If anything, it seems like the females with a high intake actually gain weight. And even though it's not really significant, this observation stuck with me, especially because so many females report initiating cocaine usage to control their weight, and then they struggle stopping cocaine use because of the weight gain that will push them back into relapse. So that's why we decided that this was interesting enough to look a bit deeper into, and we do that by looking at leptin. The reason we look at leptin is by this little fellow, or actually big obese mouse, and he's obese because he has a mutation for leptin. Leptin is a hormone produced in the adipose tissue, and the more adipose tissue you have, the more leptin will be produced. And so leptin interacts with the hypothalamus to reduce food intake and increase energy expenditure. Now, another obese mouse line actually was found that had a mutation in the leptin receptors. So leptin receptors obviously are found in the hypothalamus, but that's not all. They're actually present throughout the brain, including the nucleus accumbens and the VTA, which are part of the mesolimbic reward circuitry. And so increased levels of leptin will actually reduce the reward for food and with it food intake. But it doesn't just work on food, it also works on drugs and substances like cocaine. And so increased leptin levels have been shown to reduce the reward for cocaine. On the other hand, if you take cocaine, that has been shown to reduce leptin levels. And so there's this reciprocal inhibitory interaction. I added this little question mark because there is some controversial findings in the literature. And so this forms a very interesting stage on which our HS rats can provide some answers. For example, if we look at our high, what effect does leptin have on our high versus low addicted rats? Or on the other hand, if we look at high addicted versus low addicted rats, how do they differ in their endogenous leptin levels? And so to answer the first question, instead of sacrificing one cohort of animals that went through this behavioral testing, we added some additional experiments to look at the effect of leptin on cocaine seeking. And there's two ways you can look at cocaine seeking. One is while the rats are actually taking cocaine by adding one of these PR sessions, which is these sessions where the rats have to press more and more to get the next reward. And so we added 2 sessions, and we did that so we could do an within subject a randomized treatment of vehicle or leptin with an IV injection 40 minutes before the session. Now, the other way you can look at it is when there's no more cocaine present. So through a reinstatement session, after the rats have been through forced abstinence for 6 weeks in their home cages. So a reinstatement session consists of putting the rat back in the self-administration cage, and then they will press for cocaine, but they actually don't get any. Now, a downside of this is that over time, reinstatement will reduce because the rats will realize you don't give them any cocaine, and so they will extinguish that behavior. So because of that, the design of this experiment is slightly different, and we decided to do 3 reinstatement sessions separated by a week of abstinence in between. And the first reinstatement session, we want to see that the rats reinstate. In the next reinstatement session, a week later, we want to see the effect of leptin on the reinstatement. And then the third session is actually a control to make sure that whatever effect we see is due to leptin and not due to this extinction effect. So these are the results, and that shows that leptin significantly reduces the amount of cocaine infusions that the rats took during this progressive ratio experiment. And you also see a significant reduce in the number of lever presses that the rats make during this reinstatement session. And it is specific to leptin because in the control experiment, the responses go back up. Now, if you want to look at high versus low addictive rats, we see that there's actually a significant difference between those groups, which is a nice reinforcement of our addiction index. And then we also see with ANOVA that there is in the progressive ratio, there's no interaction, which means that the leptin has a similar effect on the high and the low addicted rats. With the reinstatement, on the other hand, there is an interaction. So leptin has an effect on the high addictive rats and not really on the low addictive rats. So that goes to show that leptin reduces cocaine seeking in cocaine taking animals, and especially in the high addicted rats. Now, all these rats have endogenous leptin levels. So how do high and low addicted rats differ in those levels? So because of that, to figure that out, we looked at the plasma leptin levels in abstinence. And because leptin is dependent on the sex, we split them up. And also leptin levels depend on body weight. And so what you expect to see is that animals with a higher body weight have a higher leptin level. And you expect to see this correlation as you see in males. However, what we see in females is that this correlation is lost. And if anything, the trend is even reversed. And that might be due to the influence of cocaine, a history of cocaine taking and dependence. So to look at that, we decided to look at the weight normalized leptin levels in correlation with the animal's addiction index, but we see no correlation at all. And since cocaine intake interferes with the leptin levels and makes any interpretation difficult, we then decided to go dig in our biobank and actually pull out samples from the animals, so blood samples from the animals before they took any cocaine. And when we then look at the plasma leptin levels at this baseline, they actually do correlate with the body weight as we would expect. And somewhat to our surprise, the weight normalized leptin levels also correlate with the addiction index, meaning that animals that at baseline have higher levels of leptin seem to be somewhat protected against the development of addiction-like behaviors later on in their life when they start taking cocaine, which is in agreement with the effect that we saw by exogenous leptin levels given to these rats. Now, the effect is small, and if we split up the sexes, we actually lose some significance in females because of a lot of statistical power. But nevertheless, it emphasizes the crucial role that leptin plays in the brain reward system. And so in summary, we show that exogenous leptin levels reduce cocaine seeking, especially in the high addicted rats, and also that baseline endogenous leptin levels have a small protective effect against the development of addiction-like behavior. So more leptin actually protect against substance use. But when you do use substances, it seems to disturb the endogenous leptin levels, especially in the females. And this could be what is contributing to the weight gain that we see in the high intake females. And finally, I want to put out a plug for our rat addiction biobanks. So we have samples available from from characterized animals, both genetically and phenotypically. So if you are in need of some of those samples for a study, let us know. And thank you for your attention.
Thank you very much, Dr. Karate. We have time for some questions, but I have a question. I was struck by your previous data about the gender differences. What would you suspect happens to leptin signaling? Let's, hypothetically, if you were to suppress estrogen or estrus hormone or give androgynous hormone to the various sexes?
That's a good question. And that's part of what makes hormones and this so complicated. They're influenced by so many things. And so seeing a sex difference was not that surprising because leptin is influenced so much by sex hormones too. I feel it's very hard to make a prediction about what will happen if you take away the sex hormones. But one thing that would be interesting and could be happening is if you would do an ovarectomy and get rid of the female hormones. Maybe the females would look like the males, and so they would lose weight and they would be less disturbed by taking cocaine, and maybe the higher addiction and the higher intake that we see in females and the higher dependency that we see in females would be more normalized and similar to males.
Thank you. There's a question in the chat, I'll read it out, from Marcita Kalupi. Sorry if I'm screwing up your name. Are you planning to do the same study with naturally rewarding substances like sucrose?
That would be interesting, yeah. I mean, sucrose is very related to the food reward, right? So I'm thinking what that would show. So, I mean, leptin definitely has an effect on food, because that's the whole start putting point. So we would assume that the effect that we see of leptin would have the same effect on a food reward, because it's food and substances. And then how, yeah, so we can follow up on that. And but we would expect, so there's an overlap between the non-rewarding food rewards and sucrose rewards and the rewarding drug rewards.
Great. Do we have any other questions? Okay, thank you very much for a wonderful presentation.
Thank you.
We will introduce our next speaker, just a little bit ahead of schedule, which is great. Mark Sanderson Cimino is a fifth-year PhD student in clinical psychology in the UCSD SDSU Joint Doctoral Program. He works with Dr. Bill Kremen at the Center for Behavioral Genetics. His dissertation is focused on improving detection of cognitive decline in older adults. He's interested in the use of longitudinal study designs to identify protective factors for normal and abnormal aging. He'll be presenting his latest data in his talk, Failure to Account for Cognitive Practice Effects, Delays Detection of Mild Cognitive Impairment, Implications for Public Health and Clinical Trials. Welcome, Mark.
There we go. It would help if I turned the screen on. Okay. Thank you all for coming. My name is Mark Sanderson Samino, and I'm a fifth-year graduate student in the doctoral program for clinical psychology. Before I start, I want to thank everybody for coming, and I thank especially my collaborators, because there are quite a few of us, as well as all of the participants who contributed the data for today's talk. There were quite a few of them, and contributed quite honestly, like a ridiculous amount of data over many years to this project. So what I'll be talking about today is some work I've been doing on practice effects on neuropsychological testing. And really my main goal for this presentation is to demonstrate that practice effects are more than just this kind of like esoteric issue that some neuropsychologists care about. And they really are kind of this pre-processing step that longitudinal studies involving cognition should do, particularly if they're doing work with cognitive aging or dementia. So practice effects are a very well-known phenomenon where individuals score better on cognitive testing at their follow-up visits. This observed improvement is really thought due to familiarity with the test or familiarity with the testing environment, as a kind of actual improvement in true ability. And a key feature of most practice effect definitions, because they do vary a little bit, is really focus on that improvement at follow-up. There really needs to be a higher follow-up than the baseline test. And as you can see in that figure, while that is undeniably a practice effect, the thing is, can occur if performance does not improve at follow-up. So here are two situations where observed performance, which is that blue line, does not improve over time. Actual ability, the orange line, is declining, but that decline is masked by a practice effect, the green arrow. Although most people don't talk practice effects in this way, we do think it's important because at follow-up, if there was no effect, if individuals were taking the test for the first time, their scores would be lower. It's just that they're being masked. So we like to say that practice effects are masking true decline. These situations are particularly an issue because of how we diagnose mild cognitive impairment, or MCI. MCI as really kind of an early stage of dementias like Alzheimer's disease. And the way we diagnose MCI is by looking to see if an individual scores below on cognitive tests. The thing is that threshold is stagnant for baseline and follow-up visits. So if there is a practice effect, it can potentially push people above that threshold. This is really an issue for those people who seem like they're declining a little bit over time, but staying in that unimpaired range at follow-up, because they might actually be in the MCI range. It's just that they're being misdiagnosed as impaired. So practice effects are getting more attention in recent years, with quite a few papers talking about them. So some meta-analyses, reviews that really do a good job of outlining different ways somebody can consider practice effects. Something to pay attention to with these papers, though, is they focus purely on regression-based approaches and change scores, which are definitely viable and useful methods, particularly looking at cognitive trajectories. The thing is, these methods really are not the best equipped to calculate practice effects if there's a decline in scores over time. We don't comment on or change individual falls below an impairment threshold, for example, MCI. And that's because that delays detection of MCI. And we're really trying to diagnose MCI as early as possible, because early detection means clinicians can get involved earlier and mitigate more decline. And then more on the research and identifying MCI early is important for identifying any risk factors or any kind of preventative research for dementia. So in contrast to those other methods, they'll be speaking and called the replacement participant method, which can affect when someone crosses an impairment threshold and is able to calculate practice effects during cognitive. It's a little different because it relies on the recruitment of a second sample called replacements, who are demographically matched to returnees at the returnee follow-up. So what you end up with is two samples that are really similar, especially on age. The only real difference between those samples is that one has prior cognitive test returnees, and the replacements do not have prior cognitive testing. So what you do is you take their test means and compare them, and that yields a practice effect. And then you, in the end, have a practice effect for each test, which is a constant, and you subtract that constant from each participant's raw score to get a practice effect adjusted score. So Dr. Ellman and colleagues used this method in a longitudinal cognitive aging study and found that even after 6 years from baseline to follow-up, there were significant practice effects, even in the presence of overall cognitive decline. They also found that adjusting for practice effects significantly increased the prevalence of MCI at follow-up, which is great. I think this is very neat. However, if you look more at what they did, essentially what they're doing is subtracting a constant for these scores, keeping the threshold the same, and then saying more people fell below the threshold, which makes sense. But the issue here is you can't really decrease MCI rates with this method. So we don't really know if these practice effect adjusted MCI diagnoses are real, or if we just kind of threw math at the problem and this kind of popped out. So we really need to validate these change in diagnoses, which brings me to what I'll be talking about today. The work I'm presenting sought to use a replacement method to determine if practice effects significantly impact MCI incidence. This would mean that failing to adjust for practice effects is really delaying diagnosis of MCI, for what I mentioned before, particularly for Alzheimer's disease, because the pathology behind Alzheimer's disease begins like a decade or decades before the onset of dementia. I also wanted to validate those diagnoses with biomarker positivity. So because the pathology for Alzheimer's begins much earlier than the dementia diagnosis, and we would expect people who would progress to MCI to be biomarker positive of higher levels of biomarkers at baseline, specifically amyloid beta and tau proteins. And then finally, I wanted to quantify the impact of practice effects on clinical drug trials. And really, this would kind of affect any study using cognitively determined diagnoses. Here, if this works out, the hypothesis was that failure to adjust for practice effects is costing studies because they're forcing studies to recruit more people, and it's costing them power as well. So for the study I'll be talking today, one of the studies, we did it with Alzheimer's Disease Neuroimaging Initiative, which is a very big, popular Alzheimer's disease study. We had about 700 participants. We were 75 at baseline, about 76 at follow-up, which was a year later. And importantly, they were all cognitively normal at baseline, and we were looking at progression to MCI as our main outcome measure. So the hiccup in this study of many hiccups was that ADNI did not recruit replacement subjects that I really wanted to use wasn't particularly viable, which was fine. Because to get around that, I identified what I've been calling pseudo-replacement subjects. And these are already enrolled subjects. They were just a subset of baseline participants who are then matched to returnees in age and other demographics. As a result, before you end up with two samples, returnee sample that's taking the tests and a replacement sample that's taking the test for the first time. Do the same type of comparison to get the practice effect. And I did that for measures, and I used baseline data for 3 biomarkers, 80 biomarkers. As a reminder, all participants were cognitively normal at baseline. We're really concerned with who converted to MCI at follow-up. When we look at our data, we did this twice, first using data without effect adjustment, and then we did diagnoses using data that was adjusted for practice effects and looked at the difference in cases. As you can see here, as predicted, there were significantly more people diagnosed as MCI at follow-up when adjusting for practice effects, specifically a 26% increase in overall MCI. The next, we tried to validate that with biomarker data. So here we're displaying folks who were biomarker positive baseline and who converted to MCI at follow-up. You can see that as predicted, more people who were biomarker positive converted to MCI when used, significantly more people, when using the practice effect adjusted diagnoses. Adjusting for practice effect significantly increased the number of biomarker positive MCI. Kind of on the other side of that who stayed cognitively normal. So we have people who are biomarker positive at baseline and stayed cognitively over time. And we find that adjusting for practice effect significantly lowered the number of people who are biomarker positive and cognitively normal. In conclusion here, from this first study, our results support those of Dr. We found that practice effects increased incidence of MCI. We're detecting MCI. The baseline biomarker data also validated our diagnoses, since we would expect more people who are biomarker positive to progress to MCI as well. And in the end, we kind of concluded that practice effect adjustment improves diagnostic accuracy, which again, I think is really cool. And I'd like to think some of the other neuropsychology folks here agree, but people with different backgrounds may be kind of wondering what in the world this has to do with clinical trials. So to demonstrate how practice effects impact clinical trials, we modeled a hypothetical drug trial with conversion to MCI as the outcome. And the drug effect was defined as a % reduction in conversion to MCI. For analysis, we did power or sample size analyses for how many people you need to recruit to be powered at different effect sizes. And we completed analyses twice, first using the unadjusted MCI conversion rate in ADNI, and then using the MCI conversion rate for practice effect adjusted data. And we specifically looked at people who are cognitively normal and amyloid positive at baseline. Here we have those results. We have the results in blue there as the standard the standard PE unadjusted diagnoses, and that is the PE adjusted diagnoses, the practice effect adjusted diagnoses. On the chart there, the x-axis is effect size with a bigger number meaning a more effective drug. And the y-axis is the number of participants you'd need to recruit to be powered at 0.8 given an alpha of 0.05 at that effect size. As you can see, the red line is lower than the blue line really across the board, meaning that you need to recruit less participants when you use practice effect adjusted diagnoses. To make this a bit more concrete, we looked at real world recruitment data from the A4 study, which is investigating whether anti-amyloid therapy can delay cognitive decline. So for those of you not as familiar with Alzheimer's disease research, this is one of the major drug studies for Alzheimer's right now. Importantly, this study recruited only amyloid positive, cognitively normal individuals because these folks are considered to be at a really, really early stage on the path to Alzheimer's disease. If we take a look at the recruitment data, they needed almost 7,000 people at baseline testing to identify who's cognitively normal. And they gave imaging to about 4,500 of those folks to identify who's amyloid beta positive. And they had a final baseline sample of about 1,323 participants. So working backwards, this means they need to give PET scans to 3.3 times their final sample and cognitively screen 5.11 times their final sample. So we took these ratios and combined it with that power chart I showed you a second ago. And we wanted to look at how many fewer participants they'd need to recruit if they had used practice effect adjusted data and had MCI as their outcome variable. So using that chart, we found that the study was powered for an effect size of 27.4. And when we look at how practice effect adjusted data lines up on that chart, we find that it actually only need 1,045 people if they use practice effect adjustment. That's almost 300 less people. Working backwards, that saves about 1,000 people on PET imaging, and about 1,400 people on initial screening. I don't work in any kind of clinical drug trial, so we reached out to some people who did. And we found out that saving about 1,000 PET scans leads to a saving about $4.7 million. Regarding those initial screenings, that's an additional $3.3 million. A lot. That said, if you were to do a true replacement method, you do need to recruit additional participants. So we estimate about 600 people at a price tag of $600,000, which does reduce savings, but it's still almost $7.5 million in savings if you did things just a little bit differently with your study and your study design, which is a lot. So I did want to focus primarily on the clinical trial data for this presentation, but we have completed similar analyses with an MCI baseline sample and found basically, in short, that adjusting for practice effects increases diagnostic stability kind of across the board here at the baseline with an MCI sample. We've also demonstrated practice effects on cognitive screeners as well. So at the end of the day, this research is the importance of practice effects. And although they're getting some attention recently, they're still really largely ignored in research. And we kind of see them as a pre-processing step that's really quite easy to do and has really big downstream effects. So by raising diagnostic accuracy, you can detect cognitive impairment earlier, which is great for clinicians who can then get involved earlier and maybe mitigate more decline. On the more research end, it improves biomarker cognition concordance in keeping with the AD, current sort of AD trends in research. And together, this dramatically reduces costs of clinical trials, which is great for clinical trials. But I think what we're all really concerned with here, regardless of our specialty, is all of this taken together really kind of improves our ability to take care of people with Alzheimer's disease. And that's really all I have for today. So I want to thank you all for listening. And yeah, thank you. I can just leave this up, actually.
Thanks so much, Mark. What a great talk. I'm sure we'll have a lot of questions, but I have one for your consideration. What do you this raises a lot of really important questions. There, a lot of folks, when you see them in the clinic, they report, subjective cognitive deficits, and then of course, as a part of routine care, often that you see clinicians use serial exams like mini-mental, slums, etc. Do you think this could possibly explain the delta between the subjective lived experience of cognitive impairment and how folks do on these tests, possibly with repeat effects?
Yeah, so comparing subjective cognitive planes to cognitive screeners, I agree, there's definitely some issues there. I would say that with serial screeners, this would be an issue. On work we've done with the MMSC, it was interesting, people who are cognitively normal did not show a practice effect, but people who are MCI did. A pretty decent one too, about a point, which doesn't sound like a whole lot, but given you only need to miss, 6 points on the MMSC to be impaired, that is kind of a big deal. So I think this would really impact that. One of the many factors that could impact why those don't always match up well.
Hi, this is Igor Grant. Can I ask a question?
Please.
Yeah, sorry. First of all, great talk. And the practice effects story is of great interest, obviously, to our group as well. I work a lot with Bob Heaton, who's been very, very interested in this issue. Maybe I missed this because I got disconnected briefly, but did you talk about just the practical aspects of kind of doing repeated testing in order to, get rid of the practice effect? And, what just the feasibility of that is either in clinic or in designing the clinical trial?
I did not. I have a lot to say about that, maybe more than we have time here, but some people do use sort of this repeated baseline measure where you get tested twice and then use the second baseline as the true baseline you use for your study and things like that or in a clinic. That has been shown to reduce practice effects. I do think that kind of method is quite costly because you have to test everybody twice, both in time and in just money to pay the clinician to do that. The other issue there is, I think this is more of a broader issue with the field of practice effects, is I talked a lot about how I think it's really important to look at practice effects in people who are actually declining over time. And I don't think that's been done by many people at all. So I think the literature doesn't really look at those situations. So as far as removing practice effects with repeated testing, I wonder if those studies are only saying practice effects are going away because there's no longer an increase in scores. I think they might actually still be there for some studies or some situations. It's just we're not catching them with the methods we're using.
Yeah, and I think, a related approach is to look at norms for repeated testing. And as you probably know, again, Heaton and his group have published on that, so that as you keep testing people, the norms you apply change rather than using norms pretending that the person is naive to that test.
Yeah, I think really the end goal here is to get practice effect corrected norms for many of our tests across some clinically relevant intervals.
Yeah, and those now are existing for at least some tests.
Yeah.
Thanks, but this is very a great talk and a great reminder about an important problem that we often don't think about.
Thank you.
Yes, thank you, Mark, for letting us know who do clinical trials. We're all in a great bit of trouble. So thank you for that. Much appreciated. We'll move on to our next speaker. Our next speaker is Dr. Miranda Francoeur. Dr. Francoeur is a postdoctoral researcher in the Department of Psychiatry, works with Dr. Dakshin Ramanathan in the Neural Engineering and Translation Lab. Dr. Francoeur is an advocate for women in science and currently serves as a voting member on the Chancellor's Advisory Committee on the Status of Women. She received her PhD in psychology from the University of New Hampshire, while working with Dr. Robert Mayer, studying the importance of thalamocortical networks and executive functions and decision-making. Currently, Dr. Francoeur is investigating how neural circuits underlying cognitive behaviors are impaired with brain injury or in neuropsychiatric disorders. She's excited to be with you today to discuss neural correlates associated with traumatic brain injury. Please welcome Dr. Francoeur.
Thank you for the introduction, and thank you everyone for being here today. Today I'll be talking just briefly about our findings with neural signatures that look like they're mediating impulse control after traumatic brain injuries. In 2014, 155 people in the United States died each day from a traumatic brain injury. Even those who do survive may face cognitive, motor, or emotional impairments. And indeed, the behavioral impairments seen after injury are very similar to those that we see with neuropsychiatric disorders, including disorders like depression, anxiety, and addiction. Impairments may last temporarily or be present for the rest of their lives with patients who have traumatic brain injuries. Indeed, about 10% of people with a TBI do develop persistent deficits. TBIs are typically classified on a scale from mild to severe. This might also explain the vast spectrum of impairments that we see after injury. Research in humans is inherently limited in its ability to understand the causality of such symptoms. Therefore, the use of animal models becomes necessary to better understand the neurobiology seen post-injury. Impulsivity, or acting prematurely without consideration of future consequences, is a central feature of many of these psychiatric disorders and is a common symptom seen post-TBI. This simple study by Cole Vonderhaar and Catherine Stanley looked at rats' performance on schedules of reinforcement in both TBI and wild type or sham rats. This experiment found no difference in behavior when the reward schedule was fixed at 1, 3, 5, 10, or 20 lever presses required for reinforcement. But when the reward schedule was variable with an average of 1, 3, 5, 10, or 20 presses needed for reinforcement, there was differences between the two groups. TBI rats had lower overall response rates, but their rate was more rapidly increased when more presses were needed for reward. These results seem to be explained by a few things that are going on. One may be that the rats were doing percevative responses, which would indicate impulsivity and poor impulse control in this task. It also could be due to the inability to adapt to the behavioral change that's necessary in the variable over the fixed reward condition. And that would suggest an impairment in behavioral flexibility. So here we aim to identify neural correlates associated with chronic brain injury in TBI animals, which may lead to these deficits we see in behavioral inhibition and behavioral flexibility. And we're going to study that by looking at changes in local field potentials recorded in awake behaving animals. For this study, rats first received a severe bilateral frontal TBI from cortical controlled impact, or CCI. With this method, injury is created by driving an impactor, shown up here, into the brain, and it's designed to mimic cortical tissue loss, subdural hematoma, or even concussion. The severity is influenced by the size of the impactor, the speed, and the duration of impact. In this study, we used a 5mm diameter tip at a rate of 3 meters per second to create a severe frontal TBI, and that's been justified by numerous other studies. This coronal section of the rat brain is just showing you a diagram of the potential damage we created at this site, our 5mm wide and our 2.5mm down depth of where we targeted the center of the injury. Rats recovered for 1 week before the second surgery, in which we implanted our probes to record local field potentials. Wild type rats in this study never received this first surgical procedure, only received this second surgical procedure that I'll describe now. Our LFP probes include 8 separate cannula, shown here again on coronal sections of the rat brain. And in each of those cannula are 4 electrodes that are each cut to different depths. Therefore, we can target 32 unique brain areas all in one hemisphere. So 8 of our cannula are shown here, and you can see the different depths of the wire, the red indicating the target brain areas that we're trying to hit. In this study, we compared brain activity in wild type, N equals 11 rats, and TBI, N equals 14 rats, as they perform a probabilistic reversal learning task. After recovering from surgery, rats begin training in an operant box that's equipped with 5 nose ports and 5 motors to deliver water rewards through the nose ports. A cartoon graphic of our box is shown here with the 5 nose ports, and then a top-down view is shown in this video. In the PRL task, rats must choose to respond in one of 2 nose ports. They start a trial with a nose response into the 3rd or middle nose port, and then have to choose either between the 2nd nose port or the 4th nose port. One of those nose ports is defined as a high probability or target reward port. In this case, in this diagram, it's randomly assigned, but here let's pretend that nose port 4 is assigned as the target. So if the rat selects that nose port, 80% of the time a water reward will be delivered. 20% of the time it will lead to no water reward. They have a chance to collect their water reward, and then the trials reset with another response into nose port 3. The other option, in this case nose port 2, is defined as a non-target. In this case, only 20% of the time a water reward is delivered, 80% of the time no water is delivered. So exactly opposite of the target nose port. Rats must perform 60 minutes of this, or they'll time out at 400 trials in each session. We have 2 different versions of the PRL task designed a little bit differently for means of analysis and electrophysiology. One of those tasks is a fixed task in which nose ports will reverse their probabilities between target and non-target after a fixed set of trials. Here's 75 trials. We also have a dynamic version of this task, which is dependent on the rat's behavior. So the nose ports will reverse contingencies after 8 of the last 10 trials are correct. Correct meaning the target nose port is selected. And I have just a quick video to show you of a rat's behavior on this task. This is a wild type or control rat performing this task. And I've gone ahead and labeled which nose port, target or non-target, he's selecting, and then if that's rewarded or not rewarded. The house lights will flash when there's no water delivery as a signal to the rats that it was no water, but you'll also be able to see that. So here he's selecting that nose port 4, which is the target, and he's been rewarded for those trials, so he's sticking with that nose port. A reversal has just occurred. You'll notice there's no cue to the rats that a reversal has occurred. They just have to figure it out based on the reward being delivered or not delivered. So he's again going to try nose port 4, but this time that's the non-target and no reward is delivered. To perform the PRL effectively, rats must respond appropriately to correct feedback when they're rewarded at the target nose port or when there's an error at the non-target nose port. But due to the probabilistic nature of our task, there's also misleading feedback that they need to learn to ignore. So you can see after a few tries and nose port 4, he switches to nose port 2, which is now the new target, and he's rewarded for those choices. So we do see significant differences in performance on the PRL task between wild type and TBI animals. Namely, in the dynamic version of our PRL task, wild type rats perform more reversals, an average of 3.4 per session, compared to TBI rats, which have an average of 1.5 per session. This effect really becomes noticeable after about 11 sessions, which is roughly 4 weeks from starting the PRL task. The rats receive no pre-training on this task before surgery. Everything is done after surgery, after the TBI is implemented. And at about 11 sessions, this is roughly 8 weeks since the TBI surgery. So there does seem to be some chronic nature to the deficits that we're seeing on this task. We can also examine things like wind stay behavior, which is the propensity to choose the same nose port on the next trial following a reward. The rat in the video nicely demonstrated some of this behavior for us. And there's also a difference here between injured and non-injured rats on the fixed version of the task. Lack of reversals and non-optimal wind stay behavior could be due to an inability to persist through the winning trials. This would suggest that animals are less sensitive to evaluating reward outcome and they're unable to update their behavior accordingly in order to correctly decide when to stay versus when to switch on those nose ports. And again, those are deficits that we see are exacerbated across time. Recording from 32 brain regions simultaneously provides us an unbiased sample of brain activity across different networks, including cognitive networks, motor, reward, limbic, visual, and even default mode networks. Activity for each electrode, shown here, 1 through 32, is time locked to task events. In this case, time from the start of the trial is shown at 0 milliseconds, and plotted for different frequencies. First, we looked only at wild type animals to find neural correlates that are associated with optimal performance on the PRL task. This is just one way to look at these results, but looking at beta activity on target rewards, we see an increase in beta power when reward is delivered. So around this time, and we see that for reward related regions, which kind of makes sense. The peak of this activity actually occurs specifically at orbital frontal cortex and anterior insula. And so we thought that might be a good place to start looking for more specific neural correlates related to this task. These regions are known to play roles in both behavioral inhibition and flexible responding. And so we kind of started with orbital frontal cortex and examined that further. We found reward-related beta activity that is greater for targets compared to non-targets in the initial and subsequent reversal blocks. So looking at target reward versus non-target reward, again, during this reward feedback period, we see heightened activity specifically to the beta frequency, and not so much during this period, at least at alpha or theta activities in only the orbital frontal cortex electrode. This activity is selective for target rewards, the high probability nose port, and does not occur for error trials. I'm not showing that here. But this indicates a match between reward expectation, perhaps, and the outcome that is actually delivered. So a match between those two things. And this signal does shift after a reversal happens. So in our fixed task, that reversal happens at 75 trials, and we can look at the activity pre-reversal and post-reversal, and we see that indeed this neural signature in OFC at beta does reverse with those contingencies. So not necessarily tracking the nose port, but really tracking the target reward. Another type of neural signature that we find is identified in medial prefrontal cortex, and this is activity that is greater for error-related activity, specifically at the theta band. This activity does not happen for rewarded trials, only for errors, and is specific to non-target errors, again occurring at theta during this reward feedback, here an error indicator. Our findings support previous work suggesting that OFC is important for developing internal representations of reward probability, where medial prefrontal cortex is important for tracking errors. These signatures may mediate optimal win-stay behavior and allow rats to ignore misleading feedback on the PRL task. Now that we've identified neural signatures associated with performance on the PRL, we want to know how these compared between TBI and wild-type animals. So looking at these two neural signatures that I just presented briefly, we do see a significant decrease in power for OFC reward-related beta when comparing target and non-target responses. And similarly, we see a decrease in medial prefrontal cortex era theta power when comparing non-target to target responses. This is a very large data set, and there's still so much more that we need to analyze, including network level analyses to see how these two potential hubs connect to the other brain areas that we've recorded from. And also we want to look at trial by trial analyses to look more at what's happening surrounding the reversals in this task. Despite this, we can already draw a few conclusions. One is just that recording from local field potential simultaneously in 32 different brain regions is feasible with CCI, and this provides a novel approach to researchers studying neural signatures associated with chronic impairments. Second is that we do see behavioral differences between these two groups on the PRL task, and these do seem to have a chronic nature to them. And lastly, we've suggested two possible neural signatures at specific brain regions and frequencies of reward and error processing that are disrupted with severe frontal TBI. These could potentially be a therapeutic target for remediating the deficits seen post-TBI. And just a big thanks to my supervisor, Dakshin Ramanathan, and the rest of the team at Neat Labs, and of course, our funding sources. I'm happy to take any questions. Thank you for your attention.
Thank you, Dr. Francoeur. We have a couple of questions. We have one question from Dr. Jared Young, which I'll let him ask, and also one from Dr. Eiler. Go ahead, Jared.
Thank you, Yash. Great talk, Miranda. Thank you very much. I know how difficult this work is to do, and so it's really impressive the amount of work that you got done, and looking at it and getting to see those differences. It was also really nice that your talk followed from the previous talk, talking about practice effects and looking at the behavior across time.
Right.
I have many questions, but I don't want to take up everyone's time. Obviously, initially, you didn't see any differences in initial reversal learning.
Mm-hmm.
You only saw differences really as the control animals began to learn to serially reverse, which is different from within session reversal learning that's referred to as, when it's conducted in humans. So, and I know there's different brain regions that subserve serial reversal learning versus just within session reversal learning. So I'm wondering whether you think that the differences you're seeing are not because it's necessarily within session reversal learning, but they can adapt to what you see in reversal learning to serial reversal. It's a shift from orbital frontal to striatal responses. And you think that do you think that they just have difficulty making that shift?
I mean, the chronic nature that we see of these impairments is definitely something that can't be ignored, and it's very exciting. I think that that's absolutely possible, and this is something that we're hoping to maybe detect with more network level analyses, looking at what else is connected to this orbital frontal structure. And based on what we see, we can actually probe if that was the same on like week 1 in both of these groups versus later weeks where we definitely see these impairments develop. So I think with our set of data that we have now, that's certainly possible to investigate in a way like that. We also are looking to extend this to different tasks. And so that might be something where we can actually look at different types of reversals and what's going on. Some of our collaborators have been really at the forefront of developing different tasks with TBI and even trying to fix performance with like cognitive cues in these tasks and things like that. So is there something that we can actually do behaviorally to improve the performance of the TBI rats on these reversal tasks? There's a lot that can be done with this behavior, but we are hoping to see some of that in this data set, absolutely.
Yeah, that's great. I mean, like you could do a progressive ratio to get it with the reward sensitivity immediately thereafter. It'd just be good to, it'd be great to, I'd love to follow up and chat with you more. So, but it'd be great to see like when the target Wednesday deficits, whether they're seen in certain time periods or whether it's like as early on or whether it's late or whether it develops over time, all that kind of thing. But I know that's a heck of a lot of work, but it was really nice. Thank you.
Yeah, we're up to it. Thank you. Love to chat more.
Thank you, Dr. Eiler.
Well, Jared stole my question, but sort of. I was also going to ask about kind of the longitudinal pattern, because what it looked to me like in your graph was that basically it wasn't that they sort of developed an impairment, but that they didn't develop the sort of aha moment that happened at day 11 in the wild type rats. So I think the focus on what is happening for the wild type rats in terms of their neural processing at, after day 11, that allows them to shoot up in their performance that is not happening in the TBI rats, I think is the crucial thing, which is what you were sort of just talking about, looking longitudinally and looking at networks. because that seemed to me to be the most striking thing, is that they just kind of stayed the same and they never had that all of a sudden like, wait, now I can do this consistently over time.
Yeah, exactly. Their reversals just stay at that level that happened early on. And we elected in this to give them no pre-training at all in any of these tasks, which is kind of interesting. And other labs have done that differently and kind of shaped behavior before giving the TBI. And so we weren't quite sure when the wild types would even, like you're saying, catch on and have this moment to reversals. So it was interesting to see that develop over time too, because that was unknown to us going into this.
Thank you very much. Thank you very much, Dr. Franco, once again. In order to make sure we're totally active for the next series, please everyone take a brief Zoom break, get up and stretch, prevent DVTs, and then we will meet and start promptly at 9:35 with Brandon Weiss. Thank you. Okay, I hope everyone had a moment to stretch, take a break, grab a drink, what have you. This time we will start with our next presenter. I'd like to introduce Brandon Weiss. Brandon Weiss is in his final year of doctoral study at the University of Georgia. He's interested in investigating the long-term effects of psychedelic compounds in personality, psychopathology, and cognition. Brandon is currently engaged in clinical training at the UCSD San Diego VA Consortium, where he's specializing in the treatment of PTSD and dual diagnoses. Additionally, Brandon also collaborates with investigators at Imperial College London. While not publishing multiple articles, Brandon can be found collecting data in South America, investigating long-term effects of ayahuasca ceremony on depression and PTSD among military veterans. Please welcome Brandon, who will be talking about changes in personality following the shamanic ceremonial use of ayahuasca.
Great. So is that right? Is that the correct screen?
Yes.
Yep. Okay, great. All right. Well, my name is Brandon Weiss, and I'll essentially good to be with you. And I'll now share kind of our research over the last 5 years examining changes in personality in relation to the use of ayahuasca in shamanic healing centers. So first I'll say a little bit about ayahuasca. So ayahuasca is known to be sort of the combination of two sets of plants, one, the Banisteriopsis copy vine, and then the second being essentially NN-dimethyltryptamine containing plants, including the two to the right. Within the kind of Western biomedical metaphysics, we understand that Banisteriopsis copy contains alkaloids including harmine and harmaline, which are monoamine oxidase inhibitors and essentially neutralize enzymes in the stomach that otherwise would neutralize the NN-dimethyltryptamine in the plants to the right, but let kind of free to pass through the blood-brain barrier. They find kind of receptor agonism at the 2A serotonin receptor site, and primarily the posterior cingulate cortex. I also wanted to share kind of the Shipibo shamanic ontology of healing as well. And Shipibo shamans were really principally the shamans at the healing centers that we recruited at. And so within this worldview, ayahuasca is a spirit and has sort of an energy, as do all plants. And that ayahuasca really aids in this healing process during ceremony. It also facilitates awareness for pasajeros and also shamans in awareness of other spirits imparting dirty or calcified energies that are understood to sort of be with human beings and organisms outside and within ceremony as well. These energies or spirits are understood to be blockages to health. And the shaman will essentially for years diet certain master teacher plants that in their kind of ontology, they become allies and essentially help the shaman to attune to these energies and spirits so as to guide positive spirits in during ceremony, open up portals, etc. And I encourage people to use the cultural references at the end if they wish to deepen their knowledge in this area. So in our study, we basically surveyed people at 3 time points: 1 week before they entered into retreat participation, immediately after the retreat participation, and then 3 months follow-up. We asked our participants to answer these surveys, and then we also invited them to essentially ask people who were significant others who knew them very well in order to generate some informant data and support our self-report findings. We recruited also from 3 ayahuasca healing centers in Central and South America. And our sample size ultimately was 256 participants who completed all time points. This was 80% of valid respondents within our self-report data, and then 110 participants were available within our informant data. Our main outcome of interest was 5-factor model personality, which includes openness to new experiences, conscientiousness, extroversion, agreeableness, and neuroticism. And sort of we feel like our results have implications also for the study and treatment of psychopathology as well, given sort of recent data-driven models like HiTOP demonstrating overlap between many dimensions of psychopathology and these domains, with the possible exception of openness. So with the awareness that this was a naturalistic study with possible sample bias and no known placebo effects, no control group, and no placebo condition, we took pains to try to place measures that would optimize the validity of our sample. So these are a couple examples of that. Our sample was predominantly male, Caucasian, also ayahuasca naive. So this is a density distribution of participants in our sample. By and large, individuals who we recruited did not have previous experience with ayahuasca, but they did have psychedelic experience. As you can imagine, retreat goers coming into an ayahuasca healing center are probably likely to have had exposure to other psychedelics before. Skewed a little bit younger, a pretty nice distribution of socioeconomic status as people were growing up. And then lower educational attainment, before undergraduate. Also wanted to just note the red line indicates the kind of sample mean within our study, and the others represents kind of normative personality standing in other samples, undergraduate and community. We found that in our sample, there was perhaps an elevation in neuroticism. And then also, I think what distinguished our sample was, and perhaps the population of ayahuasca retreat goers, is elevated openness, which is consistent with other findings showing that openness is associated with interest in psychedelics, psychedelic use. So we find that there. So now we'll go into kind of our main effects. So this is changes in personality. We hypothesize a change in neuroticism, extroversion, and openness based on previous work. Within our self-report data, and I should say we use linear mixed effects models to examine these changes. And in this particular context, this was like a repeated measures ANOVA. We were really looking for significant differences between time points. Our results indicated in the self-report data significant increases in openness, extroversion, agreeableness, conscientiousness, and a significant decline in neuroticism. The decline in neuroticism was the most substantial and meaningful at 0.62 standard deviations in baseline scores. So that's a standard Cohen's D. The informant report was really important to us because it helped to avoid some of the limitations of self-report data, potentially Hawthorne effects, placebo effects, expectancy effects, and just various biases as being part of the study. We found in this data that the neuroticism effect sort of survived within informant report data. We found here that essentially people who knew these participants well reported that they changed 0.39 standard deviations in baseline scores. We also found a significant increase in openness within the informant report data, but this was a little bit less meaningful in terms of the size of the effect. And because our audience here is within psychiatry and concerned about mental health and the relevance of neuroticism to mental health, I thought that we would highlight that outcome. This is just a nice little plot showing individual trajectories of people in our sample. And so now we'll go into the moderation-based analyses, which are really central to our study for various reasons I'll go into. So we had 3 sets of moderators: validity moderators, predisposing factors, and experiential factors. We used validity involving suggestibility and expectancies of favorable change as controls for confounding sources of change. You could imagine people who expect to change, they may report that they changed. So we wanted to control for those. We did find in this instance that suggestibility and expectancies did significantly amplify change in personality for neuroticism, extroversion, and conscientiousness, but this effective time or personality change remained after controlling for these moderators. Then we had predisposing factors and experiential factors. Predisposing factors describe sort of what people are bringing into the experience. Individual characteristics, baseline personality, whether they had previous experience with ayahuasca and psychedelics previously. And then experiential factors, what happened during that actual experience that they had. We use these to try to look at promotive factors conducive to adaptive changes in personality, and also possibly risk factors conducive to maladaptive change in personality. We also use experiential to point to, but not necessarily demonstrate, psychological mechanisms of adaptive change, even though we are doing moderation and not mediation, so we can't kind of make that jump. We also use these moderators to try to account for variability that we found within the pre-existing literature that I'll go into. The pre-existing literature is marked by small sample sizes, lack of power to perform moderation analyses, and quite a bit of variability. So I'll just share some of these results. So with the mystical type experience, we're talking about and this is sort of the MEQ subscale that's been used in the literature. So that was factor analyzed essentially into 4 factors or subscales. Mystical experience is marked by unit of consciousness, or the experience of fusion of personal self with a larger whole, non-duality, sense of sacredness, ego dissolution, We have positive mood, which is a sense of our awesomeness during the experience, transcendence of time and space, so kind of an alteration of percepts of time and space, feeling in some sense as one manifestation sort of outside of time or in a state of timelessness. And then ineffability, which is essentially this sense that the experience cannot be described adequately with language. So we find here, this is another density distribution, that the vast majority of our participants endorse sort of this ineffability, also this positive mood over the course of their ceremonial experiences, a little bit more even distribution of alterations of time and space, and moderate to extreme standing on mystical type experience. We also, as part of this study, developed a new measure that we wanted to capture sort of what may be unique about ayahuasca experience specifically. So we used, we created a quantitative measure based on qualitative data from open-ended participant reflections and then also dialogue with the Shipibo shamans and ayahuasca facilitators. We submitted a list of items to shamans and facilitators, and then we had them evaluate and revise that list accordingly. We then factor analyze all those different items. And at the right, you'll see, and three factors emerge: clarity, reappraisal, and discomfort. At the right, you'll see items associated with the clarity factor. And these are the items that loaded on that factor above 0.6. So feel free to read through those. This was a factor, and you can see the density distribution for the kind of the pink distribution at the left. So this was a factor that overlapped almost isomorphically with mystical type experiences, the other subscales that we've already discussed, but might include some facets that are interesting and kind of flesh out that construct, such as clarity and centeredness. Reappraisal was kind of a fruit of this work for us. A lot of factors within this, or facets of this within this, seem to overlap quite a bit with psychotherapeutic constructs across cognitive behavioral therapy and acceptance and commitment therapy. These include sort of an alternative perspective taking, meta perspectives on the self, confronting difficult past experiences, engaging in cognitive restructuring of different thoughts. So as one example, I felt gratitude for lessons that I learned from people who had wronged me, new positive meaning from past trauma, these sorts of things. So perhaps this construct represents something like a cognitive flexibility or a plasticity within the experience that could be conducive to change in psychopathology. And then the third was discomfort. And this was something that is commonly experienced and reported among people who experience ayahuasca. We didn't find within our sample that the majority of people were experiencing these items on the right, but certainly it does occur, and so wanted to look into that. So now to our findings. So first, we did create kind of a superordinate factor through factor analysis of mystical experience. This is sort of a catch-all for the 4 subscales of the mystical experience questionnaire and most of the AEI, the ayahuasca experience inventory that I've talked about, with the exception of the discomfort factor. And so what we found here is that neuroticism did not make it more or less likely for individuals to have a mystical experience, to have a strong mystical experience, as you see in the baseline. But those people who did report having stronger mystical experiences during their ceremonial time exhibited a larger decrease. So that's the green line in neuroticism, okay? And so specifically, we found people who are at an elevation of 1 standard deviation above the mean in mystical experience exhibited a unit change in neuroticism on the 5-point Likert scale, strongly disagree to strongly agree, of 0.54, versus those people who experience a mean level mystical experience exhibited change at 0.44 units. We also found, and I thought this was notable, that AEI reappraisal that we've talked about just now, that was the strongest moderator of change. Now, in this case, neuroticism actually did predict that did make it more likely for people to have this kind of reappraisal, or at least report having this reappraisal experience. Interestingly, those people who came in, who had higher neuroticism, had this reappraisal experience, ended up reporting at post lower, or at least as low, neuroticism as those people they came in with who had lower neuroticism to begin with. So just a really interesting effect, and perhaps there aren't statistically significant differences between those groups at post, but something to look into in the future. So we also looked into shamanic elements as well that I just don't have time necessarily to go into here, but we looked at experiences such as the medicine was cleaning me, and then also I felt that I was purging out an aspect of myself, which is something that exists within kind of the shamanic, metaphysics or phenomena that you hear from kind of shamans and pasajeros as well. And we find a similar sort of pattern of findings here, where people who had stronger experiences of these things reported greater change. We also examined whether these explained unique variance in change in personality by undertaking a multivariate analysis where we included multiple moderators, including those mystical type experiences that we previously talked about. And we found that these two, among others, or not among others, just these two did explain unique variance over and above those other subscales, which is just interesting to think about. And then finally, I just wanted to share with people kind of a comparative analysis of the effect size change that we found in our study versus other studies, other psychedelic assisted therapies, and then also psychotherapy in general. So here, I'm also working on a meta-analysis. And so this is pretty much all of the studies with one exception that have looked at personality in relation to psychedelic experience. And we're finding pretty good convergence, potentially, without a control group, of course, in change in neuroticism. And this is, just so people know, the effect size here is essentially the post-mean minus the pre-mean divided by the standard deviation in the pre-scores on personality. And so, and this is compared to sort of our effect size here in this study. And then I also wanted to share with people our results compared to meta-analytic work, meta-analytic effects of psychotherapy on personality traits. In this case, it's neuroticism or emotional stability from the Roberts meta-analysis in 2017. So we find from the first three bars at the top, ours is at the bottom, so that third bar. And this is sort of a comparison of effect size change in our sample at termination. So the second time point in our sample compared to termination within those other studies that were meta-analyzed. And then I also looked at the effect size that Roberts reported for 6-month follow-up, which is more considerable than ours, but I just wanted to give people a view of that.
Thank you, Brandon. We have to wrap it up.
Okay, yeah, and that was it. So yeah, so I'm just leaving cultural references here for people who are interested. And then thank you to my UGA team, to all of you for listening, participants and study partners.
Really fascinating work, Brandon. We have time for one question. If folks are interested, I'm sure many people will be reaching out to you because this is really interesting work.
Yeah, happy to keep going if we want to keep on time.
Is there a time for a question?
Just one question really briefly. Yeah.
Okay, this is Mariana Turner. Great talk and very interesting. I was wondering, I missed the length of follow-up and I wondered if there were plans for a further follow-up to see how much this sticks.
Yeah, thanks for asking. So the length of follow-up was 3 months, and we wanted to do that, but we didn't have the foresight to enter that into our IRB initially. And so we actually don't have IRB approval to reach out again, even though, yeah, there's some regret in there.
Can do an amendment. Great work.
And now, yeah, we've looked into it, and perhaps it's something that we could pursue further. If you have advice about how to undertake that negotiation, that would be great.
Thank you, Brandon. All right. We now have Rachel Lepidus, who is a pre-doctoral psychology intern at the UCSD Eating Disorders Center and the Medical Behavioral Unit at Rady Children's Hospital. Rachel received her bachelor's in psychology from Occidental College and master's in clinical psychology from the University of Tulsa, where she continues to pursue her doctorate. Rachel's passionate about providing evidence-based treatment to individuals with eating and feeding disorders and associated comorbidities. Rachel's research interests include exploring abnormal interoceptive processing and in the development and maintenance of eating disorders, and examining the utility of interoceptive exposure in the treatment of eating disorders. Welcome, Rachel.
Thank you so much. So my name is Rachel, as was just said, and I'm here today to present my project, which is a portion of my dissertation that I conducted at the institution before I was at UCSD, which is the Laureate Institute for Brain Research. And this project is entitled Augmentive Interoceptive Exposure Training for Food-Related Fear in Anorexia Nervosa. And this is a proof of concept study. Okay, so as we know, fear at mealtime is a hallmark characteristic of anorexia nervosa, and it's very difficult to modify. And this fear and anxiety surrounding eating and surrounding mealtime is particularly problematic, as we know that heightened meal anxiety is related to consumption of less calories at meals, and a big portion of recovery from an eating disorder and treatment for an eating disorder is weight restoration. And without weight restoration, we really know that it's not possible to cognitively or emotionally recover from an eating disorder. So this pre-meal anxiety, which we haven't been very effective as a field in treating, really poses a big threat, not only to meal plan adherence, but also to weight restoration and recovery. So as I mentioned, our field really hasn't been as successful as I think we would hope, especially in the treatment of adults with anorexia nervosa, in creating effective treatments that really target this issue. So I'd like to just take a step back and look at a field that has been incredibly successful in treating anxiety disorders, and look at the three components of anxiety that are identified in like a cognitive behavioral context. So we have thoughts, behaviors, and physical sensations. And within eating disorder treatment, we have treatments that target cognitive processes, such as cognitive behavioral therapy enhanced, and also that target behaviors, such as, again, cognitive behavioral therapy enhanced, as well as food exposure and habit reversal training. But what has largely been ignored is this third component of anxiety, which is physical sensations. And that's what I want to focus on more here as a potential treatment for this pre-meal anxiety in anorexia nervosa by targeting physical sensations of anxiety. And this, again, has been done pretty successfully for panic disorder using interoceptive exposure. So interoceptive exposure is a behavioral treatment in which individuals learn to tolerate physiological sensations through fear inhibition learning. So we can see some pictures here of an individual who is engaging with interoceptive exposure. He's running up the stairs to increase his heart rate. He's breathing through a straw to create sensations of dyspnea and shortness of breath. He's spinning to create feelings of dizziness, and these sort of feelings that might be similar to feelings that one has when experiencing anxiety or when experiencing a panic attack. And there has been some investigation of interoceptive exposure in eating disorders, although it has been pretty limited. But what has been found is that in a very small trial, that using interoceptive exposures in eating disorders more broadly reduced overall anxiety, as well as increased distress tolerance. I do want to point out, though, a significant limitation of interoceptive exposure as it is, which is that participants really maintain predictability and controllability over the stimulus that they're being exposed to. So, for example, in our patient here, he can really stop, running up the stairs when he wants to, or he can slow down, and that will really can reduce those enhanced heartbeat and respiratory sensations that he's experiencing. Whereas in actual anxiety or experience of panic in the real world, it feels really endogenous, and someone can't just decide to stop feeling those sensations, which is part of, I think, the lack of control that contributes to the exposure overall. So a unique feature that this study uses is the use of isoproterenol to engage interoceptive exposure in these participants. So isoproterenol is a beta adrenergic agonist that's an analog to adrenaline. And when through infusion of isoproterenol, you can really feel like your heartbeat is racing, you're breathing really hard, even though you're really just sitting still in a chair. So for example, when I've had an experience of an isoproterenol infusion at 1 microgram, it feels to me like I just ran up the stairs, even though I'm really sitting still. And I don't have that ability to say like, I'd rather like stop right now. It's just because it's been infused, it's just going to run its course, which is more ecologically valid and more similar to experiences of anxiety and panic that patients are feeling. This approach is also useful because isoproterenol produces a dose-dependent increase in heartbeat and breathing sensations. So we can use higher or lower doses to really augment the stimulus that people are experiencing. So that people are experiencing, which would again be more similar to different experiences of various anxiety producing situations that might happen in one's real life. So the objective of this study was to test the proof of concept utility of interoceptive exposure to target physical sensations of premeal anxiety to improve overall premeal anxiety in anorexia nervosa. So we hypothesized that this procedure, exposure to interoceptive sensations via isoproterenol, would decrease discomfort with premeal interoceptive sensations, increase ability to tolerate premeal discomfort, reduce premeal anxiety, and increase caloric consumption from a first to a second meal. And this was all within the same subjects. So our participants were 25 weight-restored female adults. Notably, the majority of our participants were Caucasian. They were weight-restored, and participants were a mean age of 23 years. Their diagnosis was confirmed through structured diagnostic interview with the mini, as well as additional consultation with a psychiatrist in our facility. And participants were excluded from this procedure if they had any history of cardiac or respiratory condition, an abnormal 12-lead EKG, or severe bradycardia, which we considered here to be a heartbeat lower than 40 beats per minute. And those were all exclusionary. Our study procedure took place over 2 visits, which were spread approximately 1 week apart. On each visit, 15 pseudo-randomized, double-blinded bolus doses of isoproterenol were administered at either 0.5 micrograms, 1 microgram, or saline. 3 infusions were given of 0.5, 3 infusions were given of 1 microgram, and 9 infusions of saline or the placebo were given during each day. After each infusion, feedback was given about the content of the infusion to improve inhibitory fear learning. So for example, if I were administered a 0.5 dose of isoproterenol, the infusion would start, we would wait 4 minutes for me to experience the effects of the heartbeat and breathing sensations. I would provide ratings, and then a nurse would tell me that was a 0.5 microgram infusion. And then after the infusion period, participants were presented with an 1,000-calorie meal. And they knew about the caloric content of the meal prior to signing up for the study. Outcome measures that we looked at were rated discomfort with premeal physiological sensations, rated tolerance or ability to tolerate this discomfort from premeal physiological sensations. And both of these ratings were taken prior to the first infusion block, and then after the first infusion block, as well as after the second infusion block, but prior to being presented with the meal. Participants were then asked to rate their meal anxiety once they were facing the meal. And this was all done on the following rating scale of 0, not at all, to 100, most I've ever felt in my life. Then after participants were given the opportunity to eat the meal, percent calories were calculated of what they actually consumed. Looking at so let me back up. I want to make sure that the study design is clear. So participants come in on day 1 of their study after completing clinical measures and clinical interview. So for their first true day of participation, they rate their discomfort with premeal sensations, ability to tolerate premeal sensations. We have the infusion block begins. They get 15 infusions of either saline, isoproterenol 0.5 micrograms, or isoproterenol at 1 microgram, which are randomized. After the infusion period ends, the meal is placed in front of them and they rate their meal anxiety. After and then are given the opportunity to eat the meal, and then we calculate the calories completed. And this happens similarly on the first day and the second day. So moving into results, let me orient you to this chart here. Our first time point is before that first infusion period. Our second time point is following the first infusion period. And our third time point is following the second infusion period. And each green dot represents a participant's rating, and the black bar represents the mean. And so here, participants' rating of discomfort with premeal sensations drops significantly, and we do see a main effect of time from the just after 1 experience of infusion with isoproterenol and interoceptive exposure. We also see an increase in a perceived ability to tolerate interoceptive sensations from the first to the second time point, as well as after the third time point. And we also see a decrease significantly in meal anxiety once the meal is placed in front of them. Now, we would expect that maybe a decrease in meal anxiety would be related to an increase in caloric consumption, which is what we were hoping for. However, we did within the study face some ceiling effects in that 80% of the participants completed their meal on both days. Not the same 80% each day, but we did have a really high rate of meal completion, which we were not expecting. And there were a few reasons why we think this might have happened. So one is that our research facility is associated with a treatment facility that we recruit from, and the cafeteria for the treatment facility is the same as for the research facility, which is also where we obtained a lot of the meals. So within the treatment facility, participants are expected to complete their meals completely, or they might not be able to continue with treatment. So one thing we're wondering is maybe we were limited in our response heterogeneity, potentially due to characteristics of our sample. Another possibility is that studies in the past have used more of a buffet option where participants are able to select the food they want to complete on their own, which might also provide more variability if we were to employ a similar option in the future. So what I think here is if going forward, incorporating a different incorporating different sample characteristics as well as a different meal procedure might help with this limited response that we're seeing here. As far as conclusions and future directions, we found that augmented interoceptive exposure was associated with decreased discomfort in response to pre-meal physiological sensations, increased ability to tolerate pre-meal physiological sensations, reduced meal anxiety when faced with a meal. And although we didn't see any changes in meal completion from the first to the second day, this to me just suggests a different need for approaches moving forward. Overall, these proof of concept results raise the possibility that augmented interoceptive exposure training could be a potentially useful tool for reducing food-related fear in anorexia nervosa. Now, I imagine some of you might be thinking like, well, it doesn't really seem reasonable or feasible to infuse my patients with isoproterenol prior to every meal for like 15 doses before every meal. And a lot of clinics wouldn't accommodate that either. And I'm not saying that's what this study is showing, but I think this study really lays the groundwork for future clinical intervention studies that focus on similar forms of non-invasive interoceptive exposure. So for example, here I have a picture of the Power Breathe device. This is something that's often used for athletes as a breathing trainer that can create sensations of dyspnea at different levels of intensity, whatever you program the device to do. So something like this type of device is much more portable, easily administered by all types of clinicians, and something that could be useful in interoceptive exposure that still has those same elements of unpredictability and an augmented nature, so that we can create a more ecologically valid experience of interoceptive exposure for patients to use prior to meals. I'd like to acknowledge my mentor from the Lloyd Institute of Brain Research, Saib Khalsa, for mentorship on this project, as well as the other members of the Khalsa Lab for assistance with data collection and data processing and recruitment. Thank you so much, and I'm happy to answer any questions.
Thank you very much, Rachel. I don't think we get enough, certainly I don't get enough experience looking at cool experimental medicine studies with eating disorders. So this is a really great example of one of that. And I don't know, I feel like on partial hospitalization programs, other similar milieus, you may be able to use infusions, but that's for another time. I think it's very possible. We're open for questions. We have time for one or two.
There's one in the chat.
Oh, sorry, I can't see the chat.
Oh, I can read it.
Okay, that'd be great. Thank you.
It said, thanks, Rachel. Did you notice any sex differences in the effects of the isoproterenol?
Our participant sample was completely female. So in this study, I can't really speak to that. However, there are certain characteristics of isoproterenol that we have seen, like in past studies from our lab, we have seen sex differences with the experience of isoproterenol, and also differences in BMI related to isoproterenol, with higher BMIs feeling the effects of isoproterenol less intensely, which can also be differentiated by sex. Does that help answer your question? I'm sorry, I don't. Does that help answer the question? I'm sorry, I don't have a more thorough answer with this study.
She said, sure, thank you.
Okay.
All right, thank you very much, Rachel. We'll transition to our next talk. Great talk.
Great, thank you so much.
I'd like to introduce Dr. Alessandra Porcu, who received both her PharmD and her PhD in neuroscience from the University of Cagliari, Italy. She did her postdoctoral training with Dr. David Welsh and Dr. Davide Dulces. She's currently a K99 award winner, completing her postdoctoral studies in the Department of Psychiatry. Her research focuses on the role of neuroplasticity in circadian rhythm and mood disorders. She aims to address the impact of environmental lighting on adolescent brain development to develop appropriate interventions to promote emotional well-being in adolescents. Please welcome Dr. Porcu.
Good morning, everybody. Thank you for your kind introduction. And thank you for inviting me today to present my project, which focuses on the effect on environmental lighting during adolescence on amygdala plasticity and its implication for anxiety-like behavior. So anxiety and mood disorder affect today 4 million adolescents in US. The rate of mood disorder and suicide-related outcomes has increased significantly in the last decade among adolescents and young adults. Genetic component as well as environmental factor can contribute to mood disorders. Adolescence is a developmental time characterized by change in the circadian system and sleep regulation. Adolescent sleep timing is delayed compared to adults. It is influenced by school start time. As you can see here, there is a difference between in the sleep duration between the age 12-14 and 15-17. You see that about 60% of adolescents sleep 7 hours or less during the school night. So if adolescents are in their bedroom at night and they are not sleeping, what they're doing? Well, I guess everybody knows the answer to this question. Today, 89% of adolescents has at least one electronic device turning on at night, which it can be a phone, a tablet, or a computer. And the proliferation of this electronic device with bright screens, as well as LED light, is increasing our exposure to blue light, especially after sundown. Several studies has been in human has showed that there is a association between this blue light condition with a risk of psychiatric disorder, breast cancer, and metabolic dysfunction. However, recently has been found an association between screen time and lower psychological well-being among children and adolescents. In particular, they found that an increased screen time was associated with less curiosity, lower self-control, and less social interaction. In human, it has been found that spectral quality of light activate the emotional brain, which is the amygdala. And here in particular, they found that blue light greatly activate the amygdala compared to green light. And this was also correlated with change in emotional responses. So which are the function of the amygdala? First of all, it regulates emotional responses, has a fear, anxiety, and aggression, decision-making, and memory processing. The medial amygdala is a subnucleus that actually receive information directly from the eye. Adolescence is a remarkable period of neuroplasticity in the amygdala. And humans have been found that, for example, amygdala volumes are related to adolescent affective behavior, and that the neuron numbers increase in human amygdala from birth to adulthood, but not in autism. So these are more classical example of neuroplasticity. In Dulce's lab, we study a newly recognized form of activity-dependent plasticity, which is called neurotransmitter plasticity or neurotransmitter switching, which is a form of brain plasticity in which environmental stimulus causes neurons to replace one neurotransmitter with another, resulting in change in behavior. So far in Dulce's lab, we discovered that altered light-dark cycle or stress or drug of abuse induce the loss of one neurotransmitter and the gain of another in the same neuron in response to this sustained type of environmental stimulation. So despite the widespread exposure to light at night in adolescents, our knowledge of the mechanism by which light affect our brain is still incomplete. So the aim of this study is to identify the mechanism by which light affect amygdala plasticity during adolescence, to reveal whether this altered light condition represent an early risk factor for developing psychiatric disorders, and to develop appropriate intervention to promote emotional well-being in adolescents. So to do that, we developed a new light paradigm to mimic human adolescent light exposure in mice. So as I said before, human adolescent light dark cycle is influenced by late sleep onset and early wake up time on school days. So this means that adolescents during the weekdays are exposed to at least 18 to 19 hours of light and only 5 to 6 hours of darkness. During the weekends, they can sleep later in the morning, so they are exposed to around 12 hours of light and 12 hours of darkness. So we use a similar model in mice, and this model consists of a prolonged light phase with light appearing here during the biological night phase of the mice for 5 days. Then the mice are kept in the 12-12 schedule for 2 days. And we repeat this 5 days to 2 days schedule for 4 weeks. It's important to point out here that the essential characteristic of light response are conserved between nocturnal rodents and human. And in particular, previous studies found the blue light at night induced alertness in both humans and nocturnal rodents. So first of all, we wanted to check if the medial amygdala neurons are activated by light at night. To do that, we expose the mice during the dark phase to 30-minute light pulse. Then we sacrifice them and we test the brain for a neuronal activation marker called FOS. As you can see here, there is a significant increase in the number of cells expressing CFOS in the medial amygdala in mice exposed to the light pulse compared to control. Then we wanted to check which type of neurons are activated by light at night. And we use a marker for somatostatin neurons because somatostatin is a well-known neurotransmitter that regulates anxiety behavior in the amygdala. And we found that around 40% of CFOS neurons express somatostatins. So this data suggests that there are different classes of medial neurons that are activated by light at night. So then we hypothesized that the chronic activation induced by light could have an effect on the plasticity and therefore in the emotional behavior. So we test our aberrant light paradigm on adolescent mice and for 4 weeks. We also have control mice that were kept in the 12-12 condition for the 4 weeks. After 4 weeks, we test the mice for anxiety-like behavior, and we use elevated class maze and active avoidance. The third days, the mice were sacrificed, and the brain were processed for immunohistochemistry. So we found that my adolescent mice exposed to the aberrant light protocol has an increase in anxiety-like behavior, as shown by a decrease in the time spent in the open arms and the total entry in the open arms. What was very interesting was that we found an increase in the time spent in the center. So the mice were actually here in the center of the maze, and they were putting their head down. And this has a risky behavior. For the active avoidance, we found an increase in the latency and number of failure to escape the shock from the grid of the chamber. And this is a freezing behavior, which is a readout for anxiety-like behavior. So altogether, this data confirm that altered environmental lighting induce anxiety-like behavior and risky behavior in adolescent mice. To test for neurotransmitter plasticity, we focus on 2 different neurotransmitters. We focus first on GABA, which is also a non-neurotransmitter that regulate anxiety and risky behavior in the amygdala. Here we found that the GABA expression is decreased in the mice exposed to aberrant light paradigm compared to control. We also analyze a different subregion of the medial amygdala going from the anterior part to the posterior part. And we found that the anterior part of the medial amygdala has a significant decrease of GABAergic cells, as in the middle part. And this is very important because different neurons in the medial amygdala project to different brain regions. So the anterior part of the medial amygdala and the middle part project to the central amygdala and the stria terminalis, which are also brain regions known to regulate anxiety behavior and risky behavior. As for somatostatin expression, we found an opposite effect. So we found an increase in somatostatin cells in mice exposed to the aberrant light paradigm compared to control. And we found in the anterior-posterior analysis, we found a significant increase in the anterior part of the medial amygdala. And these same neurons also project to the central amygdala and the stria terminalis, probably regulating anxiety and risky behavior as well. In line what we observe for neurons, we also found an increase in the CREB in mice exposed to aberrant light paradigm compared to control. And CREB is a nuclear protein that regulates somatostatin expression. So what we think is happening is that aberrant light induces a chronic neuronal activation, which in turn induces CREB activation that regulate somatostatin expression in the medial amygdala. So then we hypothesized that inhibiting chronic neural activation induced by aberrant light exposure will block neurotransmitter plasticity in the medial amygdala, therefore reducing anxiety state in adolescent mice. So to do that, adolescent mice were injected with the GI protein receptor in the GABAergic neurons in the medial amygdala. And then they were exposed to the aberrant light protocol, and simultaneously they receive a high-P injection of clozapine to specifically activates this type of receptor. And this activation selectively inactivate GABA neurons during the light at night phase of the aberrant light exposure. So we did this for 4 weeks, and then we test the animals for anxiety-like behavior using elevator plasmase. So we found that the mice that were expressing GI protein receptor and they were receiving clozapine were less anxious compared to mice that were also receiving clozapine. They were exposed to the aberrant light protocol, but they did not express the GI receptor in the medial amygdala GABAergic neurons. As for the somatostatin cells, we found that the mice receiving clozapine and expressing GI receptor has less somatostatin cells compared to mice that were exposed to the aberrant light protocol. And in the analysis of anterior posterior of the amygdala, we found a significant decrease in somatostatin neurons number in the posterior part and in the middle part of the amygdala. So altogether, these data suggests that pharmacological inhibition of GABAergic neurons in the medial amygdala interfere with neurotransmitter plasticity in the medial amygdala and reduce anxiety-like behavior. So to summarize what I have so far is that we found that light and night activate medial amygdala neurons in adolescent mice, that aberrant light exposure induce anxiety-like behavior and risky behavior in adolescent mice, and that there is an alteration in the number of GABA and somatostatin neurons in the medial amygdala, and the pharmacological inhibition of GABAergic neurons blocked the light-induced neurotransmitter plasticity in the medial amygdala, reducing anxiety state. So this is a scheme of what we think is happening. So the LED light is our switching stimulus that induce the GABAergic neurons to switch and start expressing somatostatin neurons. These somatostatin neurons has an effect on the downstream region, such as the central amygdala and stria terminalis, and increase anxiety and risky choice behaviors. So because the medial amygdala is an evolutionary conserving brain structure, we believe that this data has a potential to be translational relevance to advance mental health and emotional well-being in adolescents, and to understand how the light affect our brain, and in particular affect adolescent brain development. So I want to thank all the people in Dulce's lab, in particular my mentor, Dr. Dulcis, or my K99 mentors, and the founding agency, such as the National Center for Complementary Integrative Health and my CRESS Fellowship. And of course, thank you all for your attention.
Thank you very much, Dr. Porcu. I think this spells trouble for those of us who try to stay up at night watching cat videos and stuff on YouTube. So very nice talk. I think we have a question from Jared Young.
Okay.
Hi, Alessandra. That was really nice work. And to your point, Yash, I think that's why phones come with blue light filters these days. I thoroughly recommend using them. And I think Alessandra's talking speaks to that whole reason. I just wanted to clarify a couple of quick things, Alessandra. So obviously the exposure that you did to the animals is somewhat similar to what Davide's done in the past, but different in the sense that there's a weekend.
Yeah.
Are these, but the animals that you're using, are they nocturnal mice? So you're really exposing animals to blue light, but during their natural active cycle anyway, which is in contrast to humans. Is there any kind of, like, any thoughts as to why?
So there are different protocols that can be used to expose animals to light up night. We developed this because it was the most closest like to human, in particular adolescent, that have this like type of schedule that's different between days and weekends. And in what we think is good as a control, I didn't present it here, but we found that there is no difference between the total activity of the mice between the weekdays and the weekends. So the mice are not sleeping more because we are increasing the light exposure. So this is something that we can use as a control. And this is honestly the only way that we can do to expose the mice during the day dark phase, and we didn't see any difference in activity or sleep inducing in this. So yeah.
Thank you. I see you got lots of questions, so I'll let you go on. But congrats on the K99. That's a great job.
Thank you.
Great. Next up, we have a question. Do you have any evidence of what happens to BLA or adjacent areas to MEA?
No, yet. So this is like one of the aim of my K99 is to study the other brain region that receive medial amygdala projection.
I believe Dr. Perry also has a question.
Yes, so the Pittsburgh group has shown that circadian risk for sleep disorder. Can you hear me?
No.
Okay.
Now we can.
All right.
No, yeah.
So the Pittsburgh group has evidence recently that circadian misalignment increases the risk for substance use disorders in adolescents. And I think it's mediated by altering the reward circuitry pathways of the brain, which implicates dopamine. So I'm wondering if there's any role of dopamine in the effects.
Probably yes. We didn't study it yet. I focus on the amygdala because we observed this anxiety behavior. There are other groups that study like depression-like behavior, and they use on the lateral abenula, like Samatar group at the NIH. They focus more on the depression part because also the lateral abenula receive light information from the eye, bypassing the SCN. And I focused on the amygdala because of the anxiety phenotype observed in our experiment with our light paradigm. But for sure, there is also like a dopamine role for like other type of mood disorders.
Thank you. And the last question, and to counter Dr. Young, Dr. McCarthy is asking, is there wavelength specificity to your findings? Is blue light really worse, or is this general light as a speculation point?
So this is a great question. And I actually have currently a collaboration with Ecos. There is a LA company that developed light for buildings and office. And they have a nice prototype where you can selectively reduce the blue light. So we plan to actually test the difference of having the blue light or without the blue light. So this is the next step, and it's currently the ongoing working. So let's see what we're gonna find.
And maybe you'll present next year when you get the findings. So thank you very much.
Yes. Hopefully.
All right. So so it's my great privilege and honor to introduce our keynote speaker. Dr. Monica Fedorov is currently a third-year resident in the Department of Psychiatry's Research Residency Training Program. She received her MD from the University of California, Irvine, and a PhD in neurogenetics from University College London through a program sponsored by NIA. Dr. Fedorov has published extensively on the genetic regulation of neurodegenerative disorders, and among several honors, was recently selected as a winner of the NIMH's Outstanding Resident Award program. She presently works with Dr. Michael McCarthy and investigates circadian rhythm abnormalities as it relates to treatment response in bipolar disorder. Please welcome our keynote speaker, Dr. Fedorov.
Thank you so much. Can everyone hear me okay?
Yes.
Thank you for such a kind introduction, and it's such an honor to be here and present for everyone. So today I'll be talking a little bit about, again, circadian disturbances associated with bipolar disorder and lithium responsiveness. I'll be going into some methods of our study based on a secondary analysis from the pharmacogenomics of bipolar disorder study led by Dr. John Kelso, and then the results and some conclusions, some limitations, and future directions. So broadly, our main goal is to look at the relationship between lithium response and circadian symptoms in bipolar I disorder patients. As a brief background, bipolar disorder has an estimated prevalence of about 1 to 2% in the population. Clinically, it is characterized by episodes of depression, mania, and/or hypomania with disturbances in sleep, mood, energy level, motivation, appetite, and overall functioning. Typically, these changes in motivation, reward processing, and the affective state are significant features for the diagnostic criteria for both depressive and manic mood episodes. Circadian rhythm disturbances typically include disturbances in rhythmic and sleep and wake activity cycles, and again can be manifested in all phases, including depression, mania, and even euthymia. Specifically, depressive episodes are characterized by sleep disturbances, including hypersomnia, insomnia, as well as sleep fragmentation and overall decreased energy levels, whereas mania is defined by high energy, hyperactivity, and decreased need for sleep. There is quite a bit of evidence that disruption in sleep and/or activity can be a predictor of mood relapse. And this is supported by retrospective studies that show a significant association between mood relapse and circadian disruption among bipolar disorder patients ongoing in maintenance treatment. And this suggests that circadian disruption as both a prognostic indicator of mood stability and a key pathological feature of bipolar disorder itself. And here we just have a diagram showing the polarity of symptoms on the y-axis and then over time and how patients can progress from severe mood episodes of mania and depression to more subsyndromal depression and hypomania. So looking at the circadian rhythm synchronization, typically our sleep-wake cycles are reflections of the behavioral outputs of the circadian clocks in the central nervous system. The suprachiasmatic nucleus serves as the master pacemaker, and this is located in the anterior hypothalamus and synchronizes circadian rhythms throughout the body to coordinate them with external environmental cues such as light or temperature, known as Zeitgebers, and ultimately also coordinates with endogenous clocks in other tissues of the body. Any kind of desynchronization can result in sleep-wake cycle disturbances, for example, a phase delay, and ultimately mood dysregulation. Phase delay is often expressed as low trait morningness and an evening chronotype, and the predominance of the evening chronotype in bipolar disorder patients has been supported by a significant amount of genetic studies of polymorphisms encoding circadian clock genes, including PER1, PER3, as well as GSK3 beta and others. In the diagram here, we can see that there are both the internal and external cues that must work together in harmony to maintain mood stabilization. External cues and environmental factors include chronobiological treatment, such as sleep deprivation or light-dark therapy in controlled settings. The diagram also suggests bidirectional relationship between all of these variables, and this resonates with this ongoing homeostatic process. So in terms of treatment, as many of you know, lithium is typically the gold standard of treatment and first line, but only about 30 to 40% of patients are fully lithium responsive. Looking at data in both human and animal cell lines, lithium has shown to strengthen the rhythms by increasing amplitude and slow rhythm cycles by lengthening the period. This is demonstrated by the top diagram, showing fibroblasts derived from mice cell lines stably expressing the PER2 luciferase bioluminescence reporter to measure rhythms as PER2 expression. And PER2 expression is known to reflect network activity across the circadian clock. The measurements in this diagram were obtained using photo emissions. So on the axis, we have counts per second, and that was on the x-axis, we have time over the course of days, and photo emissions were recorded every 10 minutes over the course of several days. And this is work done by my mentor, Dr. Michael McCarthy, and this was back from 2015. Looking at more recent studies, so in a prospective study of bipolar I disorder patients on lithium monotherapy, the baseline chronotype predicted response outcomes 12 weeks later. And here in this diagram, we have on the y-axis the BOM score, which is a validated marker of morningness or chronotype estimation, which I'll talk about a little bit more later. And then on the y-axis, or I'm sorry, the x-axis, we have either non-responder or responder, and there's notably a significant difference in the level of morningness based on responders after this 12-week stabilization period. Finally, there have been multiple other types of studies, including actigraphy studies, which have shown that good lithium responders show high amplitude sleep-wake rhythms. And additionally, case reports have indicated that the correction of rhythm abnormalities in the context of successful treatment with lithium is feasible over extended period of study time. So ultimately, all of this data suggests that circadian factors may be a key component of therapeutic outcomes in bipolar disorder. However, it has not yet been determined or demonstrated in a prospective study that lithium corrects rhythm disruptions such as sleep and energy symptoms, and specifically how lithium affects circadian disruption and its relationship to a clinical response. So here I'll go into the primary methods of the study. As I mentioned, this was data initially derived from the Pharmacogenomics of Bipolar Disorder Consortium, which is an 11-site prospective, non-randomized open trial of lithium monotherapy for bipolar 1 disorder patients comprised of 386 subjects. So the primary goal of part 1 of the study was really to identify genetic markers of lithium response during the maintenance phase in bipolar 1 disorder. What I'll be focusing on today is a secondary analysis, which is really looking at the relationship between circadian rhythms and the therapeutic response to lithium. So briefly, we have 4 groups here among the 386 subjects as defined by the key, with one group of patients considered unstable on another medication, so not lithium, a second group of unstable but on lithium, thirdly, stable medications on lithium polytherapy, so lithium and an additional psychotropic medication, and then finally stable on lithium monotherapy. So in the first part of the study, in the cross-sectional component, there were 2 sets of baseline measurements obtained. The first was chronotype of morningness, and this was obtained via the basic language morningness scale, which I mentioned previously for chrono estimation. Excuse me. Secondly, the second set of measurements were derived from 11 items from the TEMPS-A scale, which is a scale that assesses 5 dimensions of affective temperament. These were scored as absent versus present, and specifically the questions that were of interest were those associated with sleep, diurnal mood variation, and energy, and the sum of these items were used to calculate what comprised as the circadian disruptive index, or CDI, to estimate the overall circadian disruption, with a higher number indicative of more disruption. Moving over here into the perspective part of the study, this was really the focus of these 88 subjects who were initially unstable and on another medication, and thus naive to lithium, were transitioned to lithium monotherapy over a 12-week stabilization period. And this was where we had clinical assessments to assess for relapse and ongoing mood stabilization every 2 weeks by clinicians. By the end of the 12-week period, there were 6 sets of standardized measurements obtained using modified forms of the QIDS-SR, which measures symptoms of depression, and the CARS-M, which measures symptoms of mania. And these were obtained from each cohort to determine symptoms that we made a dichotomy essentially of circadian versus affective symptoms, chorus symptoms of depression and mania. And then specifically, we define circadian symptoms of depression as associated with sleep, energy, and concentration. We define negative affective symptoms of depression as those associated with feelings of sadness, worthlessness, anhedonia, and then total symptoms of depression, including both circadian and affective symptoms, as well as those that we considered more ambiguous and did not kind of nicely fit into either category, such as appetite and weight changes, restlessness, psychomotor slowing, but were again included in the total score. Similarly, with mania, we define circadian symptoms of mania associated with sleep, energy, and concentration, and symptoms, positive affective symptoms of mania associated with euphoria, irritability, and grandiosity. Again, the total symptoms of mania included both the circadian and positive affective symptoms, and the more ambiguous symptoms not nicely fitting into either category, which included things like thought process, speech, insight, delusions, but again, were included in this total score. So looking at the first set of results, here we have a table illustrating the prevalence between individual circadian disruptive index items and morningness in each respective cohort, with the group values indicating the proportion of positive scores and group mean for each component. So first here, looking at the morningness, this is based on, again, those BOM scores, which range from 13 to 55, with a higher score indicating greater morningness. And again, with the key here, you can see that the group stable on lithium monotherapy had the highest level of morningness, and then gradually followed by the two groups in the middle, and then the lowest being the unstable on another medication. Secondly, the individual circadian index components were selected again from the TEMPS-A scale. And again, because they were scored as either absent or present as 0 or 1, this depicts the group sum of each cohort, and these were transformed to z-scores based on the baseline group to normalize for comparison across various symptoms and control for differences in their initial symptom severity. I will also note that the sample size decreased over time with some subjects as they dropped out of the study, but there was no effort to impute any missing result following the dropout. So looking in addition to the morningness, as we can see among these other traits that comprise the circadian disruptive index, there was a consistently significantly higher level in the patients unstable on other medications, and then lower in the stable on lithium monotherapy. And this was statistically significant to varying degrees, as indicated here, for 9 of the 11 items that we looked at. So ultimately, this is suggesting that lithium exposure in bipolar disorder patients is associated with fewer symptoms of circadian disruption, and that specifically any exposure to lithium, even in patients that are not fully responsive, is associated with greater levels of morningness than not being on the medication whatsoever. Look at the next set of results. We have varying levels of overall circadian disruption comprising the circadian disruptive index. And this is regarding the relationship to lithium exposure in figure A, and then secondly, in figures B and C, the interrelatedness of these specific traits. So looking at figure A, we found that the index differed significantly across each medication group, with stable patients on lithium monotherapy harboring the lowest circadian disruption index of around 3, higher and relatively similar values among the two middle groups, around 4.7, and then finally the highest level of the circadian disruptive index burden in the unstable on other medication group of around 6.1. Moving on to figure B, we're looking at several items such as rarely tired, sleeping less than 6 hours, low energy, comprising the circadian disruptive index, and looking for the degree of interrelatedness in the form of a heat map with a positive score significant above 0.2 and increasingly significant in the positive direction with a brighter red color, and then a negative significant score or negative correlation below negative 0.1, and again, increasingly negative statistically significant correlation with a deeper blue color. In figure C, we're looking at the clustering results of a principal component analysis of the first two dimensions and seeing that items comprising the circadian disruptive index tended to cluster in 3 relatively distinct groups, suggesting an interrelatedness of specific dimensions or traits with each other and unique from other clusters of interrelatedness. Specifically, the cluster names were labeled based on the traits that tended to aggregate, with the low energy cluster showing traits of low energy, often tired, yet sleeping greater than 9 hours, and with a high degree of interrelatedness. The excess energy showing traits of rarely tired, sleeping less than 6 hours, and diurnal mood variation, AM greater than PM, with a high degree of interrelatedness. And then finally, the unstable group here, having more erratic mood and sleep-wake cycle disturbances with significant fluctuation and energy shifts. So again, looking at the data from here, this is suggesting that any exposure to lithium is associated with not just the increased morningness, but also here we really have the reduced burden of circadian disruption, and that's really regardless of the direction of the symptoms. In the last set of results, we're looking at the clinical course of those 88 subjects in the unstable other medication cohort as they began lithium treatment and were followed prospectively over the course of 12 weeks with assessments at every 2-week intervals. On the right, we're just showing that there are no significant differences in baseline circadian disruptive index, morningness, lithium level, and baseline depressive or manic symptoms. And then secondly, just to note, of these 88 subjects, they actually did split evenly into 44 lithium responsive and 44 non-responsive between here and here. So figure A shows that in patients who were lithium responsive, depression was gradually and significantly reduced across all measures, which included the total, affective, and circadian symptoms. And secondly, the degree of improvement was similar for all, for all 3. However, looking at figure B in the lithium non-responsive group, we can see that the total and affective depressive symptoms also significantly improved in the lithium responsive group. However, the reduction of circadian depressive symptoms by lithium was attenuated and failed to improve significantly over time, as seen by this line here. Figures C and D demonstrate the evolution of circadian, affective, and total symptoms of mania over the 12-week period, revealing that all 3 measures significantly improved in both cohorts. So again, we can see a failure of depressive symptoms to resolve was specifically associated with lithium non-response. So the main conclusions, again, that data suggests that any exposure to lithium, even if patients not fully responsive, is associated with greater morningness and a reduced burden of circadian disruption. And that secondly, from the data in the prospective part 2 of the study in patients who were previously naive to lithium, their responsiveness directly correlates for potential with improvement in specifically the circadian depressive symptoms, suggesting that stabilization of circadian symptoms of depression may be a key feature of lithium's therapeutic effects in bipolar I disorder patients. Finally, just some limitations and future directions. As an outpatient study, we acknowledge that there was a low burden of manic symptoms among bipolar disorder 1 patients who are typically in an inpatient setting, and there may not have been a sufficiently high enough manic symptoms to detect reductions in circadian disruption associated with severe mania. Secondly, Some symptoms of depression and mania did not nicely fit into this dichotomy that I described of circadian versus affective symptoms. Thirdly, the circadian disruption index was clearly measured through indirect measures, which brings me to my final point that future and current studies have started to use accelerometry and actigraphy for greater precision, and this is by using wearable devices to measure directly and more objectively patterns of movement, sleep-wake activity, behavioral components, including food and water consumption, as well as physiological measures such as heart rate. So here's just an example of some encompassing measurements. So with that, I just want to thank first and foremost, Dr. Michael McCarthy, who has been instrumental in terms of guiding me, training me, supporting me, and as a mentor. And so a huge thank you to him. Secondly, Dr. John Kelso, who has led the Pharmacogenomics Bipolar Disorder Consortium and all the other consortium members. And then I also want to thank Dr. Yoshi, Dr. Squirtle, Dr. Light, and Dr. Daskalakis, as well as the rest of the Judd Symposium committee for this honor and being able to present today. And with that, I open for any questions.
Thank you very much, Monica. We have a few questions already. I believe Dr. Eiler has a question.
Yeah, Monica, great work. It's really sort of a two-part question, but maybe interrelated. So we know that animal studies show that lithium can have an anti-inflammatory and a neuroprotective effect. And we also know that sleep has effects on cognition and on inflammation. And so I wonder in the study whether you were able to look to see if maybe inflammatory markers might mediate any of the relationships you saw with lithium, and or whether there were cognitive effects that were related to circadian rhythm deficits.
I think that's a great question. I think mechanistically, we didn't go, this was more obviously of a clinical study in terms of symptoms. I mean, I think some of the points that you mentioned regarding cognition could somewhat be measured through some of the traits like concentration, but I think from a more objective standpoint, we didn't look at those specifically, but those would be really interesting to look at and see if there was any relationship.
Thank you, Monica. If you can answer in one phrase, Dr. Perry has a question. Is there an effect on sleep onset or offset time?
In terms of sleep, I mean, ideally the period should be overall lengthened. So essentially it kind of depends what phase you're in, but the idea is to basically synchronize everything back into a phase that's in tune with your body's natural cycle.
Thank you very much, Monica. I want to thank Dr. Fedorov. I want to thank all of our speakers today. I think we've definitely seen the range of the amazing talent we have in terms of young investigators and folks in training here at UCSD. So it's always a pleasure to be at the Judd and kind of see everybody just doing just phenomenal work. I do want to give this opportunity to make sure that Dr. Daskalakis gets the last word, but I just wanted to say, as a former presenter, everybody did an amazing job, and what talent we have. So, Dr. Daskalakis.
I echo Yash, and special thanks to you for being such a great MC. Thanks, as Yash mentioned, to the 8 terrific speakers, their mentors, collaborators, and members of all of you as a Zoom audience. This was a great Judd Symposium, my first. I've been extraordinarily impressed with the quality of the lectures, and this will not definitely be my last. While there are many aims of the Judd Symposium, A key one is to increase connectivity and collaborations across our great department, despite the Zoom format. And to do that, we're going to have to exert some additional effort to really get together in a more meaningful way, and to do that virtually. In that regard, please remember to save the date for part 2 of the Judd Symposium, which is on Tuesday, March the 16th, from 9 to 11. I just want to conclude with special thanks to Neil and your outstanding leadership in PET and the Judd Symposium. We will miss all of your exceptional contributions. Thank you for all your hard work. Thank you all again. I hope you all enjoy the rest of your week.